Good morning. Thanks for joining us. Welcome to USC for those of you who are traveling from across the country. My name is Asal Habibi. I serve as a director of the Center for Music, Brain, and Society, and I'm so delightful to have had the opportunity to invite my friends and also the scientists that I've always admired to be part of this year HCN Segil Symposium. I'm just going to give you some logistical information, and then I'm going to ask Chris to say a few words about HCN and the purpose of the symposium. The hall has two doors. Exit doors, one the one that you came through, one right here in case of an emergency. We're going to have 2 breaks and a lunch break, so you'll have time to get up and stretch your legs and use the restrooms, which can be accessible from the lobby. Other than that, we have very excellent and exciting talks. So enjoy your time. We have time for questions after each talk, but all the speakers will also be available during the breaks, so you can interact with them. So with that, Chris.
Thank you, Asal. I'd like to start just by giving my absolute heartfelt thanks to Asal Habibi and the Center for Music, Brain, and Society for organizing what promises to be a fantastic second annual HCN Segil Symposium. For those of you who don't know anything about HCN or about Segil, I thought I might say a few words. HCN stands for the Hearing and Communication Neuroscience Program, which is a training program here at USC. It's sort of co-sponsored by the Keck School of Medicine, the Dornside College for Letters, Arts, and Sciences, and the Viterbi School of Engineering. So it's a very interdisciplinary program. I guess at the moment we have 20 affiliated labs from all around USC, and they study all aspects of auditory neuroscience and vocal communication, from the cell biology of auditory development up through perception and behavior, and even up to the society and social level. And I'm very happy to announce that we've just secured, I think, our 3rd renewal for the HCN training grant. As Asal said last night, if the NIH itself survives, I think HCN will too. So I guess HCN was founded around 17 years or so ago by Sarah Botcher and Neil Segil. I think Sarah is somewhere in the audience here. Unfortunately, I guess in July of 2022, Neil Segil passed away. We decided to rename this sort of annual HCN Symposium in his honor, to recognize his many contributions to the program over the years. I think he would really be delighted with the subject of today's symposium, because in addition to all his other interests, he was really a lifelong fan of music. So with that, I guess I've been tasked with introducing the first speaker, Jessica Grahn, and she's an associate professor at the University of Western Ontario. And she joined the psychology department and the Brain and Mind Institute at Western in 2011. And she has degrees in neuroscience and piano performance from Northwestern University, as well as a PhD from the University of Cambridge. And she's received the Charles Darwin Award in Public Communication of Science from the British Science Association, the Early Researcher Award from the Ontario government, and a New Investigator Award from the Canadian Institute for Health Research, which I suppose is the Canadian equivalent of the NIH, is that right? Yeah. And her research is supported by the James S. McDonald Foundation, through an Understanding Human Cognition Scholar Award, the Grammy Foundation, the Parkinson's Society of Canada, BrainScan, and all 3 major Canadian research councils. So with that, I'm very delighted to introduce Jessica, who will speak about some aspect of music.
Lovely. Thank you very much for that nice intro. I'm happy to report I've actually made professor a couple of years ago, but clearly failed to update my website. So I'm going to remind you that, thank you very much for coming in. I do like to feel like my existence here means it was not the other Darwin Award. I always find it very funny when she's trying to explain this award that I got. But I'm telling you about this, so you must know it's not the other. Lovely. So thank you so much for having me today. It's a real pleasure to be here. I'm going to talk about my research on the relationship between timing and rhythm and movement, and a lot of that will be focused on how the motor system of the brain is or is not supporting feeling the beat in musical rhythm. So this work really stems from the observation that music seems to make us move. And we don't move to other sounds in our environment. There's a truck backing up outside my window. I don't suddenly find myself so compelled to tap along or bob my head to it. But music really seems to do this. And it does this from a very young age. So here are my little demonstrating twins. So these twins are 9 months old. They can't really talk very much yet. They can't walk yet. They're not really, doing that much in the world. Yet they somehow understand that music is something to move to. And we observe this not because, some proud dad decided, okay, guys, YouTube star time, music. This is what we do to music. We have to move up and down. None of that is required. This happens quite spontaneously. And whenever neuroscientists see something emerge at quite a young age, we start to get a little bit excited about, well, maybe this is something that the brain is really wired to do. So that behavior is what I was interested in, but I was also really interested in the neuroscience of that behavior, which requires not working with fun, interesting stimuli some of the time, but actually quite boring, isolated, but well-controlled stimuli. So the first work I'm going to talk to you about is a lesson that you should really pay a lot of attention in graduate school to the stimuli that you are developing, because you may be stuck with them for the next 20 years. So what we actually move to in the music, if you can strip away things like the harmony, the melodies, a lot of the richness, it doesn't really matter what the timbre is. The thing that we really seem to be moved by in music is the rhythm. And in particular, we have this tendency to extract a steady underlying beat from that rhythm. And if we're feeling a rhythm but can't find the beat, we often don't move at all. So the beat is really crucial to giving us a sense of what to move to in a rhythm. Even if it's a rhythm we've never heard before, if we can find that beat, then we can start synchronizing our movements to it. So I was really interested in whether this might mean, this observation that we move to the beat, that there's actually something fundamental that the motor system is doing in the timing of these types of auditory sequences. And to look at this, I conducted an fMRI study where I just decided to look at what is happening when people are listening to different rhythms. And I manipulated these rhythms so that some of them were quite easy to feel a beat in, and you get to hear it a couple times, and some of them were a little bit more difficult. I also had a third type that was even harder, but it turned out the more difficult and very difficult were really similar to each other. So I'll just play two here. So, and because the biggest problem we have when we're actually doing these studies is not that people are tapping along and jiving away, it's actually that they fall asleep, because it's a bunch of sleep-deprived, kind undergraduate and graduate students are coming to do these studies, so we have to give them a little job to do to keep them awake. And for the same reason, I'm going to give you that same job to do this morning, which is to listen to these two rhythms, and then hear a third rhythm, and determine if that third rhythm is the same as the first two or different. So here's the first one. So we're getting a little suppression there, but hopefully I'll just, oh, this is just the rhythm itself, actually, not the. So it's basically dun, da, da, dun, da, dun, dun, which most people have no problem sort of extracting as sort of dun, da, da, dun, da, dun, dun. We're not getting the full stimulus here, so the task is basically you might hear something like dun, da, 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 dun, dun, and you would say, oh, that's different than the first one that I heard. I'll play you an example of this next rhythm. It has the same time intervals, but they're reordered such that the beat isn't quite so easy to hear. Yeah, we're definitely having some sound issues there. So it's not that hard to hear, but it's just a little bit more da 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 da. So it's not impossible. If you saw it written out, you could probably figure out where a beat could be placed, but it doesn't spontaneously give rise to this pretty straightforward beat percept. But crucially, has the same timing constraints, same overall number of intervals, same length of sequence, and the actual interval durations are the same, just reordered such that that beat is not so easy to hear. And when we look at what happens when people are listening, and what we do is we analyze just these first two presentations where you aren't doing the task yet. This crucially removes the activity that we're looking at from the response that people are making, because they don't know what response they're going to make until they get that third presentation of the rhythm. So by looking just at the first two, when people are actively listening and trying to remember these rhythms, we can really isolate the activity that is in response to perception, not production. And what do we find? Well, auditory cortex, right by the ears. We also found a whole network of motor areas, the supplementary motor area out in the midline, premotor cortex bilaterally, bilateral putamen and other parts of the basal ganglia, sort of core motor structure in the brain, as well as bilateral cerebellum. Now, this was for all rhythms, regardless of whether or not you could feel a beat in it. So this really does suggest that there's something about auditory sequences that engages our movement system. One area we did not see was primary motor cortex. That's the part of the motor system that responds when you're actually moving along. We also instructed people and observed them, and these rhythms are not that exciting, so it wasn't that hard for them to not tap along. Now, what do we see when that rhythm really affords that ability to move along, when you can feel a beat in it? So here, the next image is areas where we see more response to beat rhythms than non-beat rhythms. And what we find here, and then in several studies afterward to replicate, is the putamen, particularly of the basal ganglia. Now, we also searched to try to find within these areas, like the supplementary motor and premotor cortex, was there hints that maybe they were responding differently to beat and non-beat rhythms that we couldn't really see at this whole brain level. And so here we just have a complicated graph where the purple rhythms are beat rhythms and the two shades of red are our two types of non-beat rhythms. And here we have all the subdivisions of the basal ganglia, the pallidum, putamen, and caudate, and all of them respond in a similar way where they're more interested in beat than non-beat. The supplementary motor area also shows this pattern. And in this study, the auditory cortex, that is superior temporal gyrus, showed it, but we don't find that consistently across studies. However, as you can see, the cerebellum and premotor cortex do not seem to care whether that rhythm has a beat or not. So their activation in this task is not being driven specifically by whether that rhythm has a beat or not. We went on further to look at how communication between brain areas changed as a function of the rhythm that you were listening to. And what we found is that correlations between putamen activity and between cortical motor and auditory areas were higher when you were listening to rhythms that had a beat versus rhythms that did not. So not only are the putamen more active, but they're communicating more with other cortical motor and auditory areas when it's a beat that you can tap along to. And again, this was always done in the context of no actual movement, usually just passive listening, or in the case of the discrimination task, active listening. We also looked at individual differences. So here we asked people to listen to rhythms, but before they went in the scanner, we assessed their musical training, and we assessed whether they had a good beat perception in the context of music. So we asked them to listen to little musical clips and determine if some metronome beeps were on or off the beat. And what this enabled us to do is look at areas that were responding more strongly in people with better beat perception while controlling for musical training, because this, as you can imagine, is always a thing that we come up against. And what we found here was that it's, again, motor areas that seem to be more correlated with better beat perception. It's not auditory areas. So here we have the supplementary motor area again, as well as the dorsal premotor and ventral premotor cortex. So interestingly, the premotor cortex, which doesn't seem to care very much about beat, was responding more when people were better at perceiving a beat. Now, all of these are just general, is this area more active or less active when you're feeling something that has a beat or not? But we can actually get a little bit more fine-grained. One possibility, for example, in our earlier study with the premotor cortex or the cerebellum was that the average activity across the region looked similar between beat and non-beat rhythms. But we can actually dig into that activity and not just look at activity that's averaged across a whole set of what we call voxels. These are the little cubes that we break up the brain into for analysis. So in this figure, we're going to look at multivoxel patterns. Basically, if the underlying neural distribution in this whole region, which is denoted here by a circle, differs slightly. So if certain neurons in certain parts of that region are more active for one condition than another, and we just average across all of them, we're going to see the same average level of activity across all of our conditions. But what this example shows is two possibilities. One is that, yes, if we average across all of these darker and lighter blue voxels, we get the same average level of signal, and we say, oh, okay, this brain area doesn't care about beat and non-beat. But actually, if we looked at the pattern, we would find that, well, when it heard a beat rhythm, we had this particular pattern of activity. Sorry, I'm not having the easiest time with the trackpad. And we got a different rhythm that also had a beat, and that pattern was actually quite similar. And then we got a non-beat rhythm. Oh, pattern looks a little different. Got a beat rhythm. Oh, that rhythm looks similar across to the two other beat rhythms that we heard. Oh, non-beat is looking consistent as well. So we actually miss subtle variations in the signal that tell us, actually, this area does care about whether there's a beat or not. But the way it's showing that it cares is not just global activation, it's actually in the underlying pattern. Or we can contrast that to a rhythm an area down here, which is not showing consistent similarities across different rhythms that have a beat. It does seem to care about the different rhythms that it's listening to. So we might say that this is an area that is rhythm sensitive. It is noticing that there are differences across all of the different rhythms and responding differently. And again, if we just averaged across all of those, we'd say, oh, yeah, it responds to rhythm, but doesn't care about what type. But here we might say, okay, well, it doesn't really care about whether there's a beat or not. That's not the feature that's important. But it is encoding something about rhythm. So it is sensitive to what type of rhythm we're looking at. So a lot of fMRI is moving in this direction where we characterize whether the patterns are similar or dissimilar across different types of stimuli as another measure of whether that area is actually encoding or doing some processing related to that feature. This would be in contrast to an area, for example, that we don't think is involved, in which it doesn't have any distinction across its patterns, no matter what stimulus you're giving it, or that those distinctions are completely inconsistent. You give it that stimulus once, gives you a pattern, same stimulus again, different pattern. It's clearly not involved in processing of these rhythmic patterns. So when we do this across the brain, this might give us a sense of whether, for example, the premotor cortex or the cerebellum is beat sensitive, but it's just not showing up when we do all this like smeary averaging that we often do in fMRI. So the first picture I'm going to show you here are areas that seem to be rhythm sensitive. They show consistently similar patterns to the repetition of the exact same rhythm, and they show different patterns across different rhythms. And as you can see, we see the supplementary motor area, premotor cortex. Auditory cortex now really does show this sensitivity. It notices that you're playing it a different type of rhythm. Also the cerebellum, so a lot of the same areas, but now the auditory cortex really shows, yes, it does care about what kind of rhythm it's listening to. However, when we say, is that consistency being driven by whether the rhythm has a beat or not, so that first condition that I showed you where we get consistently similar patterns for every type of rhythm that has a beat, we still only find the supplementary motor area and putamen. So there really seems to be something about this basal ganglia and supplementary motor area that care about beat specifically, beyond coding, oh, I can tell that I'm listening to different rhythms here. Whereas all of these other areas are sort of like, yeah, I can tell this is a new rhythm, but the beat isn't the thing that's really driving how I activate. So this was very interesting to us. One of the things that we also look at in rhythm perception is that we tend to average across the entire period of listening. But actually, beat perception does not just happen instantaneously. There are stages to it. So when I first start hearing a rhythm, I have no idea, is this going to be a rhythm? Is it going to be regular? Is it going to be irregular? Is it going to have that steady beat in it? So we have this initial stage, which has been given lots of different names by different researchers, of induction. Sometimes it's called finding, initiation, inference. So it's possible, for example, in our own work, that really the basal ganglia is being driven by that early stage. It's helping do the processing to find that underlying regularity. However, an important part of the beat is that we actually continue it mentally when we're listening to a rhythm. So we like to know where the beat is, and if I suddenly stop that rhythm, people are able to carry on for a little while saying, yep, that's the beat. Even though the rhythm is not here anymore, I can keep going. So it's also possible that actually what is happening in our stimuli is that we're not seeing the basal ganglia so much early on, but really it's being driven by the fact that once you have that beat, you're sort of mentally imagining it and carrying it forward. And then finally, another thing that I just think is interesting is that we are flexible in our beat perception. If the rhythm changes, your DJ is doing some beat matching as he's trying to switch from one song to the other. Maybe they don't do it very well, and so you have this awkward moment where you're like, okay, Oh, no, now we've got the new song. Okay, we can start synchronizing to this again. That adjustment period is also of interest, and there's reason to think that the basal ganglia might be involved in that because it's involved in the processing of what we call prediction error, which is if I'm predicting when that next beat should happen, and then the stimulus doesn't line up with that prediction, I'm now calculating like, okay, I'm off. What do I need to adjust to get back on again? So this is a study done by Daniel Cameron when he was working in my lab. He's a percussionist. And when you have a percussionist in your lab, suddenly your stimuli get a lot more interesting. So hopefully these will play because they are quite interesting. What he did is he created a long rhythm. Now all of these rhythms have a beat, but because of the length of the rhythm, we can now look at what's happening in that finding stage into continuation and then into adjustment. So we have 4 types of rhythms and they each have 2 lines. So you basically have 2 rhythms of 2 different pitches continuing at the same time. In the simultaneous congruent, both rhythms just start at the same time, and they both have the same, what we call meter, which is they are both having a beat that aligns at the same period in time. So these are both in 4/4 meter. They both have a beat occurring sort of every 4 units of time. So that's quite easy. They're very congruent in the beat that's being emphasized in both rhythms. However, you can also make them more interesting. Start them at the same time, but one is more in 3 and one is more in 4. They still line up together, but depending on which stream you're listening to, you might hear the beat as 4 or you might hear it as 3. Now, this is where we can also look at finding. We can stagger these so that we force you to listen to one type of grouping, either in 4 or in 3, and then we fade the other one in so you integrate it. So that might happen here. So in our incongruent condition, you're listening to a nice rhythm with 4. We fade in the rhythm that's got beats of 3. It just integrates into the perception. But then we drop out this one of 4, and you're left with the beat that's in 3, which you've been hearing all along, but interpreting it in the context of a beat every 4, and now suddenly you have to switch to every 3. So that's, I think, the most interesting one, so I'll play that one for you so you can get a sense of what these different stages of finding and continuation and adjustment are like. So I'm not going to talk too much about the incongruency effects, like what happens when you're listening to 4 versus 3. They don't really affect the basal ganglia results that I'm going to talk about. But so this enabled us to look at what's happening when you're finding that beat and you're getting it in the first place, what happens when you're continuing it, and then what happens when you have to adjust it, you have to switch it. And what we find, so these are just the different stages against rest, against silence. So on the top left here, we've got very clear auditory cortex. That's great. They're listening to rhythms. We want that against a silent baseline. But we do indeed see the basal ganglia coming online, the sort of dorsal or more high part of the putamen. When we look at continuation, we see putamen again. So again, auditory cortex quite clearly, but in a different part of the putamen. And then the adjustment also very strongly brings back the putamen, but again, in this different area. So on the right here, we just sort of plotted the areas that are responding during finding, which is in red here. And as you can see, the red is really a distinct part of the basal ganglia compared to the continuation and adjustment, which both seem to overlap in this more ventral posterior part of the basal ganglia. So actually, the putamen seems to be involved in all 3 stages, but different parts of the putamen are involved when we're finding that rhythm versus when we're continuing it or even adjusting it in the context of having had a beat already. That's not the same. That adjustment of phase does not appear to be the same as when we're finding it without some prior context. So all of this work has really, I think, solidly shown that we've got this network of motor areas that are implicated in rhythm and beat perception, and that really the basal ganglia and the SMA are very important for beat perception specifically. Different regions of the basal ganglia seem to be important for different stages of that finding, continuation, and adjustment. However, all neuroimaging is just correlations. So the next study we did, we actually modulate people's brain responses. So here we use transcranial direct current stimulation, and we can look at what happens when we make an area more excitable, where it's not like TMS where we're causing firing and disrupting. what we do is we just make that area's resting membrane potential a little less negative. So the neurons in that area are more primed to fire if relevant inputs are received. If we run the current the other way, then underneath that electrode, those neurons are more inhibited. That resting membrane potential is made more negative, and now those inputs need to be stronger to fire. And what this enables us to do is determine whether, for example, if we excite or inhibit the supplementary motor area, we improve or impair your rhythm perception. So we did this study over the supplementary motor area as well as right and left premotor cortex to determine, again, whether we would see this causal response where when we're changing brain responses, we're changing behavior. So people came in and did 2 sessions. They would get a sham session, and then one group of people would get excitatory stimulation over a particular area, another group would get inhibitory. And then we had 6 groups because they had 3 different areas to look at, and half of them get excitatory stimulation and half get inhibitory. They're doing that same task we did in the scanner, where they're determining whether rhythms are different or the same. And what we're going to look at is how good they are at detecting whether those rhythms have changed. Now, what we find for the supplementary motor area is a clear effect. So here, we have excitatory stimulation on the left, on the top for beat rhythms and for our non-beat rhythms. And supplementary motor area excitation improves performance in both of those. Down here, we have it for inhibition and for beat and for non-beat rhythms, and we can see that performance decreases reliably when we're inhibiting the supplementary motor area. The premotor cortex is a little more confusing. There's no reliable effect, but if you see those little gray lines, those are individual changes in performance, those little slopes. Some individuals are having quite strong effects, but it's not consistent. We tried to tease apart whether maybe it's their musical training that makes it more or less consistent or some other background factor. Nothing consistent came out of that either. It is possible to do TDCS where you actually acquire MRI scans and then model the current flow for everybody's individual brain structure. So maybe that's more necessary for the premotor cortex. That's another avenue we can try. So again, we find that the supplementary motor area, it's not just along for the ride, it's actually doing something important for rhythm discrimination, such that when we enhance it or inhibit it, we make people better or worse. Premotor cortex, whatever we're doing, is not consistent enough to make any conclusions yet. So it's one of those lovely null results that you get to go explore for the next 5 years and try and figure out. So this provides nice converging evidence that there's a crucial role of the SMA, and it could be that the basal ganglia, so we can't target the basal ganglia with TDCS because it's so deep. That would obviously be what I would prefer to have done. But the SMA and the basal ganglia are quite interconnected in the way that they communicate. So it is possible that it's that processing loop that's really important. And when we knock out one part of that loop, then we're really impairing people's performance. Now, I don't know about you guys, but when I go home for Thanksgiving, I have a cousin who's in the Peace Corps, and another one who runs their own company, and people who are doctors and really helping people. And they come and you say, oh, what do you do for work again? How's that going? It's I look at whether the brain, an area you've never heard of, is a little bit more responsive to this small tweak we make to rhythms compared to this other small tweak that we make for rhythms. And guess what it is? And that's nice, dear, So there's always a question, what can we do with this that actually might be therapeutic? So I'm going to talk, the last part of the talk, about some work we've been doing with rhythmic auditory stimulation. And what we're really interested in is the effect of rhythmic auditory stimulation for disorders in which motor areas are commonly affected. And Parkinson's disease is a classical example of this, where because of a death of neurons, dopamine neurons that feed into the basal ganglia, they actually have difficulty initiating movement. While they're walking, they may exhibit freezing. They have unsteady gait, their balance is off. And they may show shuffling gait as well. And we know that their basal ganglia activity is abnormal. And I'll show you an example from YouTube about why people are interested in using musical rhythm for these patients. This is a patient with this. Let's give you an idea of the walk.
Okay, so now we're going to do gait training with music. Everybody's got cell phones. Everybody nowadays has music on their phone, so this is just kind of a real easy way to do it. Let's stand here for a minute. We'll get the rhythm. Once we feel it, then we'll take off.
So very dramatic change. Great, problem solved. Let's give everybody their playlists, and Parkinson's will will be solved. The gait issues will be gone. The issue, of course, is that it's very variable. So what do we mean by rhythmic auditory stimulation? Well, scientists are very insecure. We have to go home to Thanksgiving and impress people with what we do. So instead of saying, I play music or a metronome while people are walking, we say, I'm delivering rhythmic auditory stimulation. But what we mean basically is we're playing a music or a metronome while people are walking. It's usually at or near walking pace. So often it's at walking pace or maybe 5 to 10% faster to increase movement speed. And then we measure various aspects of gait to see whether they improve, freezing, their speed, the stability or balance. No good speeding people up if you're going to make them more likely to fall because their balance is off. So it's important to make sure we're not making certain gait things worse. And when we look at this in the lab, we do see patients who look like the patient in that YouTube video. So here is a patient walking, no stimulation. They've hung a little what they call virtual doorway. So it's a curtain because that often induces freezing when people have to go through doorways. Their job is to go press a button on the wall and turn around. As you can see, the festination of gait there, the shuffling as they turn, and then confronted with this doorway, there's a long episode of freezing. And they're wearing a bunch of stuff to measure their gait. That's what all the cables are that this person is assisting with. And then they're able to go. Now you won't hear the metronome, but this is while this person is listening to a metronome that's been set to 10% faster than their baseline walking pace. So much smoother. So again, works not just on YouTube, works in the lab. Super promising. Whenever you have a patient like that, that's really striking, and clearly that's a tool that can be useful for them. But it's also incredibly variable across individuals. So often you'll have, if you look at the individual plots in a particular study, 2 or 3 patients that show a fairly dramatic observable effect, 3 or 4 or 5 that are kind of improving, but not dramatically so. And then another 3 or 4 or 5 that no one talks about who are actually a little bit worse. And so when you actually look at the mean, the overall size of the effect looks quite small. But what that's really indexing is that some people have a great benefit and some people really don't. This is caused by other things as well. Often these are small n studies. It's not very easy to ask patients to come into the lab all the time. There's variation in what people deliver. Sometimes it's metronomes, sometimes it's music. Sometimes the experimenter fancies himself a bit of a composer, so they make the best music that they think will help everybody. Sometimes people bring in their own music, which has its own variation about how appropriate it is for walking. Or sometimes there's sort of a database of music that's selected. And if you look across studies, there are often different effects on different gait parameters. So some will find that stride length is improved, and others will find that velocity is improved, but actually the length isn't that much better. It's because they're taking more steps per minute. Or some will find that gait is improved in terms of balance. So you can measure that with stride width. When people are feeling unstable, they widen their stance. And so that gives you a sense that they're feeling unstable. And if you see that width is increasing during these, that's not a good sign. It means that they're feeling the need to compensate for something. One way to improve this would actually to understand how RAS works. Now, I've just spent half an hour telling you about how, well, we know the basal ganglia responds to the beat. That's definitely how it works. We actually have no idea how rhythmic auditory stimulation works because the motor system is complex and there are lots of different pathways that feed into it that might be tapped into by RAS. If we could understand these mechanisms, then we might actually be able to decide for different people what aspect of the music is most important. So this is the brain according to Jessica. It's my box model of the brain. Basically, what we're trying to do with rhythmic auditory stimulation is increase excitability here in the cortex to improve movement. In Parkinson's, the striatum acts a bit as a brake on movement in healthy people, and this brake is on too strongly in Parkinson's. So we need to remove the brake. And there are different ideas we have about how RAS works. One is indeed the striatum just is part of the basal ganglia that includes the putamen, that listening to this music or rhythm activates the striatum, makes good use of what remaining neurons are there, and increases the overall excitability, leading to greater output throughout the motor system. However, there's a really interesting thing in Parkinson's. When they want to make voluntary movement, that's often when they have problems. Reflexes are okay. If you ask them to reach their hand out, it may take some seconds for the command to respond, and it may be slow. You throw a ball, no problem. And we think that that might be related to the fact that the cerebellum and premotor cortex, which we know respond to externally triggered movement, are preserved in Parkinson's. And so that maybe what this rhythmic auditory stimulation is doing is tapping into this preserved external cueing system and enabling people to move that way. The other possibility is that people just like music, and we like moving to music, and the nucleus accumbens, which is where the area that responds when we are listening to things that we like, has a fairly preserved dopamine source in Parkinson's, at least early on. That's not where the dopamine is low. And so maybe it's just that they like walking to this music or like synchronizing with a metronome, and that's actually what's increasing this. So a lot of what my lab is interested in is working out what's going on anatomically here. But before we can go into the brain, we actually need to understand what parts of the music might tap those different pathways. So we look at groove. How much does the music make you want to move? Groove is something that basically, I think, goes along well with that beat perception. It's the thing that makes you want to tap along. Enjoyment, that allows us to look at whether the reward pathway is very important, because you can have music that's groovy, but you don't like very much. I can't help tapping to this, but this is not something I would choose to put on. Or maybe it's that external cue, that beat salience that's really important. So how clear is the beat? So that's what we're trying to examine, is the contribution of these three different factors. And the variability we see across people may be because we're using these different factors differently, and some people may really need that enjoyment. They might have terrible beat perception. It's just not going to work for them. Or some people might be like, okay, if you give me that clear beat, then that's really helping me. So they're really using it as an external cue. So to do these studies, we have people walk on this mat. Just looks like a little carpet, but it's filled with pressure sensors. And that enables us to automatically measure, without having to wear all those cables, different aspects of their gait. And what I'll show you today, I think is velocity that we're going to be looking at. But we can look at all the other things as well. And then we manipulate musical features like groove. So we have them listen to a bunch of clips of music and say, oh, that's high groove. I really want to move to that. Oh, no, that's very low groove. I don't want to move to that. So that for each patient, they're getting their own selection of music based on what makes them want to move or not. We can also manipulate enjoyment. So is this music you like or not? And we try to do this while keeping the ratings of the other factors equivalent. So high enjoyment versus low enjoyment, but fairly equivalent on whether it makes you want to move or not, or how clear the beat is, so that we can independently manipulate these different parameters. Beat salience, what we've been doing is manipulating it by adding a metronome to the music. So you might have low groove music that you don't want to move to very much, but it's now got a sort of boom, 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 boom. So the beat is at least very clear, even if it's not music that you really want to move to. We ask people to synchronize. The data I'll show you today is all asking them to synchronize. We also look sometimes at when they're not synchronizing, and we say just walk freely, with the idea that some people who have bad beat perception, you now just give them a difficult dual task when you say, now synchronize. I know you have problems with gait, and you might not be very good at perceiving the beat. Try to do those things together. We do generally seem to find better effects with synchronizing, though, so we haven't actually found evidence of that dual cost effect for most people. So today I'll just show you the synchronized data. And then what we do is we match the musical beat to be about 10% faster than their walking cadence, so that we're not asking people to walk to something that's dramatically faster or slower than what they would walk to normally. And it means across all of our conditions, that tempo, that beat rate, is controlled. I'm going to show you data from younger and older and Parkinson's participants today. The reason we look at younger and older is, one, people who are older start to show gait problems sometimes. So that can be an interesting comparison in itself. And two, we have no idea if these mechanisms that are involved are actually different for younger versus Parkinson's patients. If it turns out that the effects are really similar, it just is the case that if you have gait problems, tapping into these mechanisms is now an intervention or a therapy, but actually it's the exact same thing that's going on in the brains of younger people, then we can do a lot more testing and piloting in younger people who don't have constraints on their time or not the same constraints on their time or on their energy, and can come in and do all of these studies while we really try and hammer out the individual differences. So what do we find? It turns out that musical groove absolutely matters, and this is true for younger and older and Parkinson's patients. So this is the change in stride velocity from baseline. So this is about a 5% increase in stride velocity for high groove music compared to low groove music. So as I said, this is true for younger people here on the left, older in the middle, and also people with Parkinson's. Surprisingly, enjoyment in our hands has almost no effect. So people say, we used unfamiliar music so that we wouldn't have a confound. So maybe that's part of the problem, is that we've kind of capped how much people can really enjoy this music. But on a 100-point scale, there's about a 45-point difference between our high and low enjoyment conditions. So it is the case that people are rating them much more enjoyable in the high enjoyment than they are in the low enjoyment, but this does not come out in their gait at all. So we don't see any difference between the high and low for younger or older or Parkinson's. And then finally, when we look at beat salience, I'm going to split it here. We've got high groove in pink and low groove in blue. And then when we have added a metronome, we've enhanced that beat salience. It's the darker color. So you can see for younger people, adding a metronome does indeed increase how much they move or how quickly they move. And it does so for low groove, but there's still quite a large groove effect here. For older participants, when we add the metronome to the low groove, it now brings their velocity up to about the level of the high groove without the metronome. So again, adding that metronome is doing something on top of the groove. With our participants with Parkinson's, that metronome actually brings the low groove right up to the high groove. So that beat salience does seem to be more important for people with Parkinson's, and equivalently important to groove. Once you have the high beat salience, the groove actually isn't that important anymore, as opposed to older and younger, where there are sort of two independent main effects there that are going on. There's the groove and there's the beat salience. So this suggests that the mechanisms may be slightly different, or at least what's important about the music may differ for what's happening for people who are older and younger versus people with Parkinson's. So we are continuing, I'll just summarize here. So groove and beat salience do alter stride velocity. Enjoyment effects are really minimal. And this beat salience does seem to be more important for patients. And what we'd really like to do is do some brain imaging. So we've been working on a couple things. One is to use virtual reality so that our environment is more realistic. This allows us to put things like obstacles to really induce gait changes that you might not get normally, but to do it in a safe way. And then we've actually been working a little bit with functional near-infrared spectroscopy, FNIRS. This is a technique that is sensitive to something similar that fMRI is sensitive to, these sort of changes in blood oxygenation. It can only be used at the surface. It just shines infrared light in through the sort of top inch or so of the brain, and when it comes back out, you can look at the alterations of how much of that infrared light has been absorbed, and this gives you measures of the oxygenation changes. But for things like the supplementary motor area or premotor cortex or cerebellum, FNIRS could work quite well. And it is not sensitive to movement artifacts the way that fMRI, we can't have people walk around at all. And even if they're doing like foot pedals, there's a little bit of movement that happens when people are moving their feet because you sort of have to stabilize yourself. So it can be tricky. This technique allows us to look at what's actually happening when they're listening to the music and it's helping their gait. So we can look at what's changing in those brain responses and start to get an idea of what these mechanisms really are. So that's everything I wanted to tell you about today. We know that rhythm perception relies on motor areas because when you change motor area activity, you actually change people's rhythm perception, and that the effects of music on movement depend on the specific musical features that you're listening to, and the importance of some of those features may differ for people with Parkinson's compared to people who do not have Parkinson's. Now, as always, I get to do the fun things like come tell you about this work, but it's other people who've done all the hard work. So I just want to call a shout out to Christy here, who has done a lot of the gait data that I just showed you. And then Marina here, who has been really instrumental in the brain stimulation data. And thank you all very much for your attention this morning.
questions. So if you just raise your hand, Scylla's going to give you the mic.
I'm very loud. So in the last study, you showed the results for beat salience was seemingly better for the Parkinson's than the older. What was the average age of Parkinson's patients versus the older?
They're matched. So, and they both tend to be in their sort of mid to late 60s as an average, with a range. I think we take people as young as 45 because we have a couple early onset Parkinson's or earlier.
Why do you think the Parkinson's benefit?
I wonder if it is this need for an external cue, that actually they can't take advantage of the groove stuff as much as the older adults can. And so that extra external cue now boosts their performance because that is really what they need. That's pure speculation, of course, at this point, but that's what we're going with.
Here. Right here.
Thanks.
Very nice talk. I just have a question about the magnetic resonance techniques that you're using. You kind of referred to that in the last part of the talk, but what is it that you're measuring there when you're doing the MRI?
Yeah, so fMRI is sensitive to the changes in blood flow that happen after brain activity. So what happens is neurons do not have their own local storage of oxygen or glucose. And so once they're active, they deplete those, they send out a signal, nitric oxide, to the local capillary bed and say, we need fresh blood, otherwise we're not going to make it. This is why if you're suffocating, your brain goes, right? The rest of your body has its own internal stores, will last without oxygen for some time, but your brain can't. That change in blood flow, there's actually an influx of oxygenated blood. And the reason we can detect that with a strong magnetic field is because hemoglobin, which is centered on an iron molecule, has different magnetic susceptibility when it's binding oxygenated blood versus when that oxygen has been removed. So when the oxygen is removed, you get different magnetic signal. So this influx of oxygenated blood actually creates very tiny changes in magnetic signal that we can detect using MRI.
Mm-hmm. And what about the time resolution of that? Would you be able to, is it that you're pinpointing activity areas, or could you detect, some sort of frequencies or the nature of the actual sound that you're sending, or is it just the areas?
No, so you have some temporal resolution where you can dissociate things that are on the order of seconds from each other. So if something's happening for 1 or 2 seconds and then something else is happening the next 1 or 2 seconds, you can definitely tell those apart. You cannot do things like 10 or 20 or 30 millisecond resolution, at least not without very clever and careful designs. And that's because the blood flow response itself is sluggish. Because the blood flow response itself is also sluggish, things like oscillations would have to be at incredibly low frequency.
You're averaging basically the whole response. All right. Thank you so much.
Sorry, do you have one more time for one question? One more question. There's one down here. One there. Sorry, multiple. I think we have time for 2.
Hello, Jessica. I wonder if you could share a high-level view of why it is humans have unique beat perception compared to other primates, because I know that's an interest you have.
Yeah, and I didn't show, we've been looking a little bit at whether monkey MRI responses are actually similar to humans, and are they showing beat sensitivity even though they don't show this movement to the beat. I think there are a couple of ideas out there. One is that it hangs onto the speech system, so that actually this evolved as something that's useful for speech, which is to be able to modify your output to match somebody else's input, and then that same machinery is being used or sort of co-opted for moving to music. There's also an idea that vocal learning is very important, so that's similar as the co-opted for speech, but narrows down specifically on the flexible changing in your speech. And so therefore we would predict species that have this vocal learning should also show this. There are also a whole range of theories about social bonding and altruism and increased cohesion that might happen through coordinated movement, and that when your groups get large, you can't coordinate movement through visual signals very easily, but auditory signals provide something that everyone can move along to. So the whole idea of why this happens is, well, it's lots of fun, but there are a few different ideas, I think, and I don't know if we'll ever know. We'll find out why we do this.
I think we have time for one more. One quick one, maybe. Niels.
Hi, thank you. I'm a bass player, and My experience is that there's a sort of with the bass, there's a conjunction between the rhythm and the harmony. And I was just wondering if you have any experience with, because you spoke mostly about rhythm, but if you have any experience with harmony and if there is a connection with that.
Yes. So in music, we use all of the cues available to us to find that beat. And then those cues also induce our desire to move. So things like harmonic stability on the beat versus transition and change off the beat are another way that when we're listening to music, we actually, oh yes, I'm right, I've got the beat here. And then creating that counter evidence, having off beats, that can actually increase people's desire to move, potentially to keep track of that beat, right? So in funk music, you got a lot of stuff going on off the beat and you get a lot of movement. So that beat clarity isn't the only thing that matters. You need enough clarity to know where to move, but then that counter evidence might be creating this urge to move as well. And I think there's some evidence that urge to move can be modified by harmonic complexity as well. If things are way, way, way too complex, we don't really like to move to it so much anymore. If it's in that sweet spot of interesting but not too complicated, that can enhance this desire to move along. If it's super simple, it's not really adding anything. So harmony does indeed play a role, as does register. Bass is really good for getting that beat and that movement. So we've shown that as well. Thanks. So you can.
Right, thank you very much. So we're going to move from beat and rhythm to imagery. Our next speaker is Petr Janata, who's a professor at UC Davis. He holds an appointment at the Department of Psychology and is also a faculty member at the UC Davis Center for Mind and Brain. He's a cognitive neuroscientist studying the psychology and neuroscience of music, especially interested in the basic psychological and neural mechanisms that underlie strong experiences that people have with music, such as music-evoked remembering and feeling. Professor Janata has served multiple times as the board member for Society for Music Perception and Cognition, and he also was the founding member of the UC Music Experience Research Community Initiative, UC Mercy. In addition to teaching and publishing scientific articles, he has given over 100 invited lectures nationally and internationally to both professional and lay audiences. With that, let's welcome Petr.
Well, thank you very much, Asal, for the invitation. Thank you all for being here. And so what I'd like to do today is to invite you to consider the idea of an auditory mental image and really sort of the central role that that plays in our experiencing of music. And what I'm going to try to develop is the idea that perhaps we should be thinking about auditory mental images in terms of sort of a distributed representation across the brain's perception and action systems. So this all begins with this recognition that music is a multifaceted process, and that even though we may just talk about hearing the music or listening to the music, that it's really not just perception in isolation of action. We may have, as Jessica mentioned, sort of the spontaneous movement that we exert with music in the form of these overt movements, which may then, we feel our movements. We may hear the sounds that we produce if we tap along, or certainly if we're playing music. But it's also important to recognize that there's all this covert action that happens, okay? And that's really the substance of my talk. It's this singing along in our minds, or the expecting of specific notes to occur, or words in a lyric. And then also here, recognizing that this perception-action cycle both helps orient our attention, can be shaped by our orienting of attention, so we can listen for specific notes, specific instruments within the musical scene. And then there are various memory systems involved, so memory for the music itself, as well as, for example, memories that we might associate with the music, so music-associated autobiographical memories. And really, our musical experience in any given moment is going to sort of draw on or engage these systems dynamically. So just a little bit of anatomical background that helps ground this entire consideration of auditory or musical mental image. This is from work by Joaquin Fuster over at UCLA, where he basically argues, hey, we can think about the brain as being divided into two parts, the entire backside of the brain. Do I not have a cursor pointer? Let's see if I can do that here. I don't usually use these. It's the very top. Oh, there we go. Okay. So the back part of the brain, which is all about perception, our primary sensory cortices, sort of hierarchical pathways for utilizing simple features, building up more complex objects, association cortices, where we build multimodal object perception. But basically, back half for perception, and the front half is really all about action across, again, various scales of complexity from really long time scale action plans all the way through the sequencing here in the premotor areas to then the implementation of the actual movements. And then also highlighted here, so aside from the hierarchical organization within perception and action parts of the brain, is the fact that we also then have communication across these areas, which I'll highlight in a moment. Here I just want to emphasize critical functional organization aspect of the frontal lobe, and it's really, we always have to consider the cortex in association with the basal ganglia and association with the thalamus. It's really is corticostriatal loops here, and the fact that they have a spiral-like organization that can promote action plans from very abstract action plans motivated by emotion and things like that, all the way through to specific actions, all the time balanced between sort of the direct and indirect pathways for either promoting or inhibiting action. So it's this very dynamic system, and really where a lot of the music cognition work is focused has been really here in these premotor regions. Okay, so with regards to some of the connectivity, a couple of bands to highlight here. The superior longitudinal fasciculus, it's connecting the lateral prefrontal cortex with parietal areas, so critical for maintaining sense of scenes. And then the arcuate fasciculus here, which also connects the lateral prefrontal cortex now with areas of the temporal lobe. And so these are going to be integral for sort of this idea that maybe these musical mental images might be distributed across some of these areas. Okay, so quick show of hands. Do you like to listen to music? All right, everybody. How about, do you like to create music? Okay, 2/3 of you maybe. Okay, so I'm going to argue that we all create music. We're all creating music all the time, obviously to varying degrees. Okay, but here's a simple example of music creation. Okay, I'm going to play you a sequence of chords. I just sort of anticipate what might come next. As opposed to. Okay, so clearly that example illustrates that we generated some sort of mental image, and in the first case, right, what we actually heard did not conform to our expectation, whereas in the second case it did. Okay, so that's a simple example, but it's really under this language of expectations from one of my first studies, ancient history here. But here's the event-related potentials measured at the vertex here, electrode CZ, classic auditory evoked potentials, some adaptation for the second and third chords. Here's that silent period, and then here's one of three possible targets. And the main focus here was illustrating that these measures of expectancy violation, the P3A component, which has a central midline generator that I'll return to in just a little bit, and the P3B, a more posteriorly distributed positivity, that really the P3B showed this graded response across the degree of expectancy violation. So violation of musical expectancies is something that consumed much of the field for a long time. Another critical location that emerged both with EEG as well as MEG recordings. Here, this response to harmonic expectancy violations in ventrolateral prefrontal cortex, so these premotor areas. Looking at something similar with fMRI, responses again in the inferior frontal gyrus, so ventrolateral prefrontal cortex, as well as the anterior insula, which tends to respond to violations of expectations really in any modality. But the idea here that when you sort of violate the expectations that a sequence of events or musical events is creating, that deviation is somehow registered in these premotor areas that are ultimately participating in these acts of sequence generation, whether they be in language or in music or neither of those domains. Okay, and so here I just want to highlight this connection, the arcuate fasciculus between these ventrolateral prefrontal areas and the auditory areas, and a really nice study by Psyche several years ago that looked at, using diffusion tensor imaging, the strength of the connectivity between the VLPFC and the auditory areas. So here in a sort of a normal music perceiving person, arcuate fasciculus similarly thick in both hemispheres. But then looking at amusic individuals, so these are people who have difficulty processing pitch relationships, okay? The arcuate fasciculus in the right hemisphere is almost non-existent, okay? And so this accords well with variety of other types of studies, multiple labs that really implicate the right hemisphere, the frontal and the temporal areas, as being important for melody perception. Okay, but what I want to turn to here is in that, for the example that I played you, right, there was that slight silent period where in the initial study we asked people to just imagine the best possible resolution. And the fact that we had to insert this second of silence was just due to a technical limitation that in the lab of Helmut Pecha in Vienna, where I was working at the time, they were analyzing the EEG in 2-second windows. And I had 3 chords that were 1 second long. I needed to do something with that extra second. So it was like, okay, imagine the best possible resolution before I presented the target, which was really the event of interest. And then when I looked at the ERPs, I was like, oh my goodness, this looks like an auditory evoked potential here when they're actually not hearing anything, right? They're just supposed to imagine the best possible resolution, which suggested to me that maybe, in fact, this act of generating an auditory mental image is activating the auditory cortex, which back in the mid-90s was an extremely controversial standpoint or point of view. You know, does visual mental imagery or does auditory mental imagery, do early sensory cortices get activated? But what do we call this, right? So in the initial framing, it's like, well, we're forming an expectation. But we could also think of it as covert orienting of attention or cued attention, right? Those first 3 chords are guiding you to expect something in a particular location of feature space, of harmonic feature space, right? So it's sort of like saying, you know, with visual attention, you give an arrow to tell people which hemifield to attend to, right? So that's the cue, right? Cued attention. And well, so the musical analogy might be you set up a sequence that generates a very specific expectation. So that's like cued attention. Or, or do we call it mental imagery, right? And so really what I want to argue is that really these three concepts may all largely be interchangeable, even though these words are used differently in different kinds of paradigms. And so if we think about this sort of more unified notion of musical or auditory mental image, we can now think about, well, what are the things that influence these representations, right? We can think about the bottom-up information, so the sensory information that's providing evidence, it's providing sort of the basis, sensory basis, if you will, for that mental image. Or, we can instantiate these images just purely from via top-down mechanisms, fetching them from long-term memory. So if I ask you to imagine your favorite song or something like that, right, you can probably all start generating those mental images, right? Those are coming from long-term memory. And then the type of task that we give a person may in fact also influence the form of the auditory mental image. So what I'm going to show you next are just a set of behavioral findings that sort of manipulate these forms of information to look at the acuity of a mental image. How is the accuracy of that mental image shaped? So the task that provides the strongest sensory support for that mental image could just be a two-tone discrimination task. You play somebody one tone, you then play a second one, which is either the same, or it might be mistuned sharp, it might be mistuned flat. They're basically making a same different judgment, right? You've got full sensory support. You're not going to be able to form a clear mental image of that tone than in this task where you're actually given the information, the sensory information. We can contrast that now with this cued attention, this expectation task, where I'm just going to give people a major scale, do, re, mi, fa, sol, la, ti, do, right? And so then they have to identify whether that final tone is in tune or not. And again, it's going to be mistuned sharp or flat by varying degrees. So we're going to get people's thresholds, right? So we're going to talk about the image width, how accurately are they forming that mental image. In this case, they actually have not received sensory information that corresponds to that image that they're supposed to form in order to make a judgment about the target. Then in the imagery task, we just take away some of that sensory support. So we just play the first few notes of the scale. They have to imagine the three remaining notes, and then for the final note, they actually hear the target that again is in tune or mistuned. And then we can further manipulate the memory support for the images by either utilizing the same key, the same major key on every single trial, so they really just build up a representation of that key, or we randomly select the key for each trial from 7 different keys, and so they're not really building up that memory support across the course of the experiment. And then we can also look at utilization of long-term knowledge about tonal structures. So in Western tonal music, here again, they're just hearing major scales. And so either we can place that target on the tonic, so the most stable note in the key, or we can make the leading tone the target, which is the least perceptually stable note in the key, and see how that might affect the images. Okay, so what we see now here in these plots, it's always two tasks plotted against each other here. The two-tone discrimination task, the dependent variable now is the image width. And so here, these participants here, the circles now are when all the trials are in one key, the plus is when they're in 7 keys. These individuals right here have an image width of about 40 cents, okay, in the two-tone discrimination task. And it turns out in the acute attention task. You hear the scale, you imagine the last one, or you hear the last one is the target, what you were expecting. So about 20 cents flat, 20 cents sharp, okay, that's our best subjects. Other people have absolutely terrible thresholds, okay? But what's interesting here is that while some people are similarly good in both tasks, some people do just well in the two-tone discrimination where they're getting that full sensory support. But now in the acute attention, all of a sudden their thresholds are somewhat higher. Okay, so their image is degrading, even just and they're listening to that scale. And overall, when you mix it up from trial to trial in terms of what key you're drawing from, there's a little bit of a cost there also. Okay, this is now the two-tone discrimination task, and now the imagery task. So we've removed even more support. Some people still do great. They're forming accurate images all the way through the notes they have to imagine, yet more people completely fall apart, and it gets harder when you're using 7 keys instead of 1 key. So just to go back, it's the acute attention. This is what I'm calling the imagery, right? So some people fall apart with the mental image even further. Slight cost when the target's on the leading tone than on the tonic, as might be expected. And then we do also see an effect of musical training, so years spent with some sort of instruction on an instrument. And so generally mental images become better with more training, although you still see people with very little training or no training who just do just fine with their mental images. Okay, so what's happening in the brain that might be shaping this mental image for pitch? So I'll show you first the results of a PET study, really the first main one that well, it was the second one, but that looked at this, Andrea Halpern and Robert Zatori. And what they did is they took, played for people melodies from familiar TV shows. So back in the time, this was Dallas. Anybody remember early 80s? Who shot J.R. Ewing? You know, that was the big question that gripped the nation. Not who would get elected, it was who shot J.R. Ewing. Weren't those wonderful days? And so they would play them the first, either listen to the whole thing or play them the first few notes and then imagine the continuation. Or then they had control melodies that were unfamiliar melodies. And in one case, people either had to repeat that in their minds or in other condition, not do anything here, all right? So they're going to now compare the imagery of the familiar melody with not doing anything during that period. And what emerges is now a network of areas incorporating both the posterior part of the brain, so the parietal lobe, in fact, the auditory cortex, as well as these frontal areas, the ventrolateral prefrontal cortex, and what is emerging as our favorite area of the day here, the supplementary motor area, okay? So the supplementary motor area being associated with sort of internally generated actions, in this case, internally generated images of pitches. But the big news here was that even though they didn't get primary auditory cortical activation, you still saw auditory cortical activation in the imagery task, sort of secondary areas. Here it's this imagery finding juxtaposed to finding from a study we did on just orienting attention within a multi-instrument auditory scene, real musical scene. And basically when people are orienting their attention in these polyphonic musical scenes, we're just recruiting a similar sort of, basically the same network, right? So either whether you're just covertly generating these images entirely, or whether it's now in the context of following along with a piece of music, it's the same frontoparietal and frontotemporal circuitry that appears to be recruited. Around the same time, I did a very similar thing. Again, little short, simple little melodies. People either heard all the notes, they heard 5, imagined 3. Here they heard 5, didn't imagine any. These were in separate blocks of trials, and this is now using ERPs to ask, is there in fact evidence of this auditory N100 potential, evidence of activation of the auditory cortex in response to these imagined notes. And so these are scalp topographies at 100 milliseconds when they hear it. Now, when it's the first of the 3 imagined notes, first of 5 imagined notes, no imagery requirement is down here, and we can see that, in fact, these topographies are highly similar when people are imagining, suggesting, in fact, its auditory cortex is being engaged. So Ana Navarro-Cebrian, a visiting graduate student in the lab, did a nice set of studies. She also did that behavioral work that I showed you. And so this is basically the paradigm I had described before. You're hearing these notes of a major scale, and then making an intonation judgment about a target, or hearing the first 4 notes, imagining 3, and then making an intonation judgment about a note that occurs when the last note would have occurred. That's either mistuned up or down. And again, we're measuring these tuning widths. So I'm going to play you 2 examples now. I'm going to play you first, hear all the notes, and I want you to tell me, just show of hands, how are we going to, well, I'll just ask you if you think it's mistuned or in tune. Who says it's in tune? Who says it's out of tune? Okay, a couple of you. It was actually in tune. Okay, now we're doing the imagery case. First 4, continue imagining, and then make a judgment about the target. Okay, in tune. A few of you. Out of tune. Okay, more of you saying out of tune. Bonus question, was it tuned sharp or flat? Who says sharp? Okay, everybody's saying sharp. Indeed, it was tuned sharp. Okay? But that's a pretty tough task, right? And obviously, as I showed you, people vary in their quality of images that they form. Okay, so here are event-related potential as just grand average across all the participants at 3 midline electrode sites. Let's just focus on CZ here. So auditory evoked potential for the first note in the scale, auditory evoked potentials for the others, this adaptation, some sort of perturbation for the first imagined note. And then here they hear a target again at the end. And so again, because you haven't been hearing anything, the response to the target is very large because it's not new sensory input that's coming in. And this aspect of adaptation is going to be absolutely critical here in just a moment. Okay, so we see a couple of these important evoked potential components. So the N100, many other studies have shown it has generators in the auditory cortex. Amplitude decreases with repeated stimulus presentation. Now for the deviants, those are the blue lines. Right? When it's mistuned, people are hearing these as mistuned on average. It now elicits this P3A component, frontal sites here, right? This detection of that expectancy violation. Area that's sitting right above the SMA and the anterior cingulate there. Okay. And then a later component, this P3B component, this posterior component. So now we can look at the scalp topographies at this these different time points. But what I'm doing now and in the subsequent slides, I'm basically doing here I'm doing a median split on the participants with regard to their imagery thresholds. So who I'm calling good performers are those who form more accurate images, as determined by their image width, and the bad performers are those who aren't forming such accurate mental images. And we see these topographies now for when they're hearing all the notes, and then in the imagery condition where they use the response to that final target. We see that across conditions, when they're hearing all the notes leading up to the target, or when it's now just the target following several imagined notes, slight difference in the morphology of the N100 component, but pretty similar across our two participant groups. But now if we look at this measure of detecting that mistuning, this P3A, we see that in the good performers, we have a clear P3A component. In the weaker imagers, let's say, it's really not as pronounced, both in the imagery condition as well as in so in the imagery condition as well as in the listen condition. Okay, so this is suggesting that they're somehow not really now detecting whether or not what they just heard is actually in tune or not. Right? It's, that sensory information isn't really getting matched against as good of, let's say, a template, an image. Okay, but here's what's really striking. So if we compare the ERPs for these two groups of participants, we can now look at the N100 in response to that final note. So following having imagined several notes. And look at this difference here. So the good performers, they have a smaller N100 than do the people who didn't form as good mental images. So this suggests that, in fact, the people who are forming accurate mental images all along are actually adapting their auditory cortex such that when they actually receive more sensory information, the auditory cortex is already adapted, and so the auditory evoked potential isn't as large. Okay, and so we can now plot this, the amplitude of the N1 as a function of image width. So here we have our better imagers. They have a smaller N100. As your mental images get worse, the size of your auditory N100 goes up. We can look at what happens with the P3A component. And so the cued attention condition, you're hearing all the notes leading up to the target, or the imagery condition. And basically, when people are forming good images, they have a large P3A than when they're not. We see these nice significant relationships. It's also present in the imagery condition, although that relationship is a little shallower. Okay, so both the N100 and the P3A appear to be pretty good markers of image acuity. And the frontal midline distribution and many localization studies localize that P3A component here to the sort of SMA, pre-SMA, and associated cingulate cortical areas, right? And so this may reflect the activity of some sort of sensory motor monitoring process that signals the mismatch between this covert action that's being generated and the sensory input that's actually coming in at that moment. Okay, there's a huge literature, in fact, on these midline sites as being involved in error monitoring in general. So there's a family of negativities, like the error-related negativity that you get when you initiate an action, realized you made an error. It's also known as the oh shit potential. There's the feedback-related negativity, when you made an action and now you get feedback about that action and it was the wrong decision. You see a negativity that's registered there, and you see this P3A component, right? So it suggests that this region is involved in when we're actively generating a sequence of actions and needing to monitor our performance. This area is critically involved in that process. Okay, so really this is, there's obviously a lot more to be done here, but this is sort of, I think, intriguing to think about this notion of a distributed auditory mental image that has components that are existing both in the frontal lobe and in the temporal cortex, and that when you're capable of instantiating a better mental image, right, that deviations from that are now going to be registered, or I guess correlates of the quality of that image are going to be registered both in the perception and the action parts of the brain. And just a little aside. So often when we do these experiments in the lab, we're just doing single trials. You have some sort of context that builds up to a target event. We analyze the response to that target event. But it's really important to realize that all our musical engagement, if we're performing, if we're dancing along, if we're singing along in our minds, this is all happening very rapidly, right? I mean, if you have like 5 notes a second, I mean, it's just 200 milliseconds. And so we're just really engaging this perception action cycle. And then we're sort of perturbing it. Like, okay, yes, it's either, the images we're forming, what we're hearing, they match really well, right? We're fluent in our experience. And so we've heard a little bit about the groove, and one conception, so that's a very broad concept, so there's the urge to move component, but another component I'm fascinated by is that sense of unity, the sense that you're sort of one with the music, or when you're playing with other people, right? When are you in the groove? You're in the groove when the sensory motor, that perception action cycle is very fluent. As soon as errors start happening, you disrupt it. And so that's why we've got a sort of emotion outlining the perception action cycle here. But obviously that's all speculation. Okay, in my last few minutes here, I'd like to just touch on another aspect of musical mental imagery that's actually very central to so many people's lives. And this is more recent work that we've been doing. Ben Cubitt, a grad student in my lab, who's now actually postdocing with Psyche. So this is this idea of involuntary musical imagery, also known as earworms or getting music stuck in your head. Show of hands. How many of you people have this experience of music being stuck in your heads? Okay, virtually everyone. Okay. How many of you find it frustrating? How many find it pleasing? Okay, slightly more on the pleasing side. Okay, I'm gonna try to offer hope for those of you who find it frustrating. Okay, I'm gonna give a hint as to how to make peace with your earworm. Okay, so earworms, many contributing factors, but they can generally be induced for recently and frequently heard music. So if you're just listening to the same song on loop, that's going to become an earworm. And I should note that it's a very prevalent form of spontaneous cognition, right? So for those people who think in auditory terms, right, what are you doing? Well, you're either running, when you're just walking across campus or something, right? You're running conversations in your mind, right? The frustrating experience you just had with your advisor or a colleague, or, what you're, anyway, lots of internal dialogues we might be having, right? Or more pleasing is like you're grooving along to some funk band in your mind, right? Okay, so earworms, prevalent form of spontaneous cognition. Well, in another line of work, I worked on music-evoked remembering, this idea that, a piece of music you hear that you haven't heard in years, perhaps, and it evokes a strong memory from your past. The question is, how does that strong association form? And one idea is, well, we know that we form strong memories when we rehearse them over and over and over again. Well, how might you rehearse memories that somehow involve music? Well, just have the music play over and over again in your mind, and that might reinforce the representation, the memory representation, both of the music and sort of the associations you've made with that music. Right, so if you're listening to the same song over and over again with your friends when you were a teenager, right, for some summer in your life, and the music's going to become an earworm, and the idea is that earworm is going to serve as a consolidation mechanism for associated episodic content. How do we test this in the lab? So we bring people into the lab day one. We expose them to 8-second long GarageBand loops that just loop over and over in the background as they're doing some sort of other task. We stitch together 4 sets of repeating loops into a soundtrack. So a soundtrack consists of 4 loops. And about 40 to 50% probability that any given loop will actually become an earworm and be reported as such in the intervening week before we bring them back on day 2. And now we associate these soundtracks with old Walt Disney cartoons. They perform an immediate recall task once we show them these 2-minute long movies so that we have a sense of how much did they retain. We then bring them back after some sort of delay period, give them this cued recall task with the music. How much of the memory did they retain? And then we're going to look at that memory retention as a function of whether or not the loop was actually being consistently reported as an earworm. So again, just day one, couple of different tasks. In one variant of this study, we used the serial reaction time task, which also implicitly engages the procedural memory system. And in fact, this sort of inhibits the likelihood that a loop will become an earworm versus just passive visual viewing of a SRT-like display. Unfortunately, the weak part in all this work is that all of our earworm assessments are just sort of retrospective self-report, and in the process of trying to develop an experience sampling way of getting better quantitative data on those. But anyway, we just do it this way right now. All right, day 2, we associate these different soundtracks now with different old Walt Disney cartoons from the 20s and the 30s. So soundtracks that were either heard in one of the conditions on day 1 or an unfamiliar soundtrack, or in this first version we used silent music. Now we tend to use scrambled music, so possible soundtracks, but now we just sort of shuffle segments of them in time to create a scrambled version. It's a bit of a better control stimulus. So just to give you an example of this, one of these, let's watch it. So you see that, and then you immediately perform, you hear the soundtrack again, and the people type out as much of the plot and the details of the movie as they can. And so then we bring them back a week later, again, play them the music, they recall as many details as they can. And so what we do then is we have these very detailed scoring rubrics for every 30 seconds that's associated with the unique loop. So we can basically tally up the details, how much did they remember this granularity of 30 seconds with a particular loop. And so then we can now plot the proportion of details that they recalled on day 3 and day 2. So a ratio of 1 would be basically remembering the same number of details as they did immediately after the movie. And what's on the x-axis is now a measure in me serial count of the consistency with which people reported in me for a specific loop. So 3 would be that they reported it on day 1 after they completed all the questionnaires. They reported it for having experienced it in the delay period between days 1 and day 2, and then again during days 2 and day 3. And what we see is there's a significant likelihood of retaining the information for those loops. This is now in those 30-second segments that were associated with loops that were more likely to be earworms. Okay, so that's really suggested to us that it's that loops-specific rehearsal of that content that's consolidating in memory. We still get a significant effect at a delay of 2 weeks, or 4-week delay with this paradigm. We don't see it. Now we have experiments ongoing to see sort of under what conditions can you get this sort of effect in terms of the pairing of the information. And it's appearing that the sequencing aspect of the unfolding narrative is actually critical in order to get the music to bind to it. Okay, so just finishing up, hopefully you're more convinced than ever that music is a rich experience that engages many systems of the brain, but centrally sort of this idea of a perception-action cycle and this notion that internal experience that we have, that mental image, can vary in accuracy, right? And that may in fact shape now how we perceive incoming information also. So acknowledge various collaborators along the way, and I thank you very much for your attention.
Thanks for an awesome presentation. I was just wondering about so going back to the unexpected harmonic change that elicits the P3A. I'm just wondering if you have a sense of how early we might see that in infancy, or if it takes a certain amount of input to build those targets, let's say.
Yeah, so that's a good question. So I should, and I think that, so Stefan Kulch, who has done this study on the early right anterior negativity, so they argue it's a sort of implicit detector of these harmonic mismatches. It occurs slightly before the P3A, so it's usually around 150, 200 milliseconds. And I think that they've looked at that in kids also, but I'm with probably Daniela Samler, maybe, I think, or Psyche, Oh, yes. With Sebastian. Yeah. So, yeah, and they find it. They find evidence of the sort of responses to the violations. One should note that they're also more pronounced in musicians, right? So again, I think it always boils down to the question of how accurately are those images being formed, and to your point, some of them form very, in kids also.
So thank you for the presentation. I have a question about like ERPs and expectation about tones or chords because people are receiving like different levels of education and experience in music under different cultural aspects. So do you think like there will be like a universal expectation or standard measuring ERP, or like it varies like from people to people based on like their system of like musical knowledge they're receiving? For example, like the Western music tonal progression like are different from like Eastern one or from Turkish one or like from the African music. So yeah, that's my question.
Yeah, with those kinds of tonal expectations, I mean, those are highly culturally dependent, and and so, but it's within any given system, musical system, one would expect to see the same ERP components, because really, I mean, the ERP components are being driven by, highly conserved neural circuitry that just has to accomplish really the same goals, sort of irrespective of, what the actual musical context is. But then I think that the amplitude of those responses is modulated by expertise with the particular system.
There's one more in the back.
Yes, thank you for that presentation. My question is, how can this be used to help with pain management? And I ask because actually I use music for my pain management during my nerve block procedures at Cedars-Sinai. They allow me to sing. I'm not a great singer. I was new to music. I joined a choir and got trained in a particular song that I love, Miserere Mei Deus by Allegri Gregorio. Amazing. It has like some soprano notes that are out of this world. And so it's music that is an airworm for me consciously, so that I can be out of be distracted from the pain of each needles. Coincidentally, some of the soprano points hit when I want to scream, so I approximate that in order to not feel the pain. I've been doing this for 7 years. Thank God they allow me to do it. I'm a physician also, but disabled now because of this chronic pain. So I'm very interested in how you can use this, and if you have any ideas for the future for myself and for hopefully patients and other doctors.
Yeah, thanks for that question. So I'll admit, I actually have not done any research on music and pain. I know there are people who are looking at that. In fact, now there are a couple of sort of NIH working groups, little consortia that are focusing on music and pain. So I expect that there to be, substantial advances in our understanding here over the next few years. I guess I will just point out that this region here, this fainter blue dot right here, the anterior cingulate cortex, is also very much implicated in pain. It's not too surprising. I mean, things that hurt us are things to be avoided, right? So it's basically an error correction signal, and so it's engaging sort of error monitoring parts of our brain. But these areas here, these midline generators, they're also really important for sort of executive attention. And so, based on your description, what I've heard from others, is that yes, if you can engage your attentional system in some way, these same areas that are overlapping with the pain systems, that might be a way of mitigating some of that. And I think it's very interesting that in your case, you're describing merging your mental representation of the pain then also with a particular timbral and pitch characteristic in the music, right? So perhaps you're somehow uniting those representations, and that's helping with your overall, coping process or management process. So those are my speculations, but rest assured that there are other knowledgeable people who are really focusing in on this particular topic. Thank you.
All right, welcome back. We're gonna get started. Our next speaker, we're going to do one more talk before lunch. So stay up for another hour, and then we'll have lunch. And after that, we'll have music. It's my great pleasure to introduce our next speaker, Dr. Ray Goldsworthy, a great colleague. Ray did his Bachelor of Science in Physics from the University of Kentucky, and then his PhD in Health Sciences and Technology from Harvard and MIT. After concluding his doctoral studies, Ray developed teams to design signal processing strategies for cochlear implants and hearing aids, and to provide auditory rehabilitation software to help people with hearing loss so they can make the best sense of music for singing and hearing in general. Ray is an associate professor of otolaryngology at USC. He's a cochlear implant user himself, and he's really interested to work on the interplay of auditory experience, auditory perception, medical bionics for improving the lives of people with loss. Let's welcome Ray.
Yeah, thank you, Asal. So it's a pleasure to tell you a little bit about my work on music appreciation after cochlear implantation. And I'm going to start off by describing what is a cochlear implant. I mean, honestly, a cochlear implant is really, it's nothing short of a modern medical miracle. And it has miraculously restored hearing to about 1 million people around the world. So how does it do that? Let me get a pointer for you up here. So there's typically a microphone or microphones that pick up sound, usually in a behind the ear unit here, picks up the sound, and then taken down to a computer chip. And for me, a lot of the magic goes on in that computer chip. How do you take sound energy picked up by a microphone and turn it into an electrical stimulation pattern? That, however that magic is done, the electrical stimulation pattern is going to be sent across the skin and skull, usually wirelessly, and brought down to an electrode array. And that's what's shown here. And so each of these sort of darker gray areas on the array correspond to an individual electrode or point of stimulation. And so each of these darker areas can be used to directly stimulate the auditory nerve. And this gets us to the, one of the fundamental problems about a cochlear implant is that the auditory nerve is comprised of about 30,000 neurons. And each of those neurons is conveying different frequency information. And so at this point in the technology of the field, the most number of electrodes that are implanted in this device is 22. So that's the most. And there's still individuals who have a single electrode being used to stimulate their nerve. And so an appropriate analogy for this is like a piano player with mittens on. And it is really true. This is the essential problem of cochlear implants is a loss of resolution, loss of pitch resolution, loss of frequency resolution. And despite that, really great outcomes can be achieved with both speech perception, but also with music appreciation and music perception. There's definitely deficits, and I'm going to go into what those deficits are. But high levels of music appreciation and music perception can be achieved. And I want to start off with what is really the most important point that I'm going to make today, and there's a big difference between music appreciation, do you like music, and music perception, how well can you hear music, how well can you hear pitch differences, how well can you hear the difference between different instruments or what have you. And so there's a big difference between music appreciation and music perception. Now, if you think for a moment, and so let me just take a step back and be clear. So I am a cochlear implant user. If I take my cochlear implant off, I'm completely deaf. And so I had normal hearing up to the age of 13, and then I completely lost my hearing at that age, and they were just doing pediatric trials in America at that time. So now when I put the implant on, I can hear really, really well. And you can imagine anybody in that situation, if you lose your hearing and you get some kind of hearing restoration, music is going to sound different. And there are deficits with cochlear implants in terms of music perception. But there don't need to be deficits in terms of music appreciation, how much you like music. But if you have normal hearing, you lose your hearing and you get a cochlear implant, there will be deficits in appreciation too. It's going to be it's like losing a loved one. You need to put in years of rehabilitative effort to get high levels of music appreciation back, like it again. And of course, that would be completely different if you were born deaf. If you were born deaf and you grew up with the cochlear implant, and that's all you ever knew, then your music appreciation could be normal from the get-go. So there's a big difference between appreciation and perception. And my lab is I've been working on perception for decades now, but we're just starting to dive into music appreciation. I'm going to give you a snapshot of that before I talk more about perception. And I want to say that along the way, I'm mostly talking about my work, but I want to kind of highlight other people who have been foundational to this work in their field. And Valerie Loui and Kate Gefeller and Virginia Driscoll has really done a lot of formative work in music appreciation, that is the psychological and sociological aspects of losing your hearing and getting a cochlear implant and rediscovering music. And I also want to point to Abraham Maslow as one of the founding fathers of positive psychology, because he was very formative to me too, and it kind of shapes how my lab looks at these psychological and sociological factors. And I think most people are probably familiar with his hierarchy of needs, where he kind of laid out, let me back up a bit again. So Maslow, what I really like about him is he took psychology away from treatment of diseases to more like, well, how can we help people flourish? How can we help you achieve your peak potential? And he kind of spelled it out that, well, of course, you first need your physiological needs, your water, food, and shelter. You need that satisfied. You need safety needs, your personal security, employment, et cetera. And then you want to belong in a community. And this is often where in my lab, when we're working with cochlear implant users and we're trying to help them rediscover music appreciation, that we come in, because we're trying to build a sense of community so they feel love and belonging and friendship and intimacy. And then building that up to esteem and then self-actualization, be all that you can be. Okay, we kind of look at that and we talk about how to look at the psychological and sociological factors and how that's going to build up and kind of make this interplay between these psychosocial factors and then perception, how well you can hear music, and then going all the way up to music appreciation. So the first thing I wanted to say that what my lab does in terms of trying to understand the psychosocial factors is we've established this cochlear implant music hour, which is a Zoom meeting that we hold almost every week, where cochlear implant users come and we bring a guest musician or a guest scientist to talk about their craft, and the cochlear implant users ask questions and are engaging. And we've been doing this for more than 5 years now. We started at the beginning of the pandemic, and we're about to celebrate our 200th meeting. And so we've met and helped more than 200 cochlear implant users through this. But we also do music in-person workshops because, of course, yes, the in-person music making is an important aspect of refinding your music appreciation. We conduct surveys of musical sophistication and engagement, and we conduct semi-structured interviews to understand the challenges that cochlear implant users face after getting implanted and losing their hearing and getting implanted and trying to rediscover music. And another framework taken from positive psychology that we use in the lab to understand this is the PERMA model, which was put forth by Martin Seligman in his book, Flourish, A Visionary New Understanding of Happiness and Well-Being. And the basic idea that, are you happy? It's too generic of a question. Happiness is too generic of a term. So he kind of breaks these down into more constructs related to the positive emotion that you feel, the engagement with an activity or a group, the relationships that you're building, the meaning and your sense of achievement coming out of that. And I'm just going to give you a snapshot of this from some of the semi-structured interviews to give you an idea of what I mean by this. So one of the participants said, I liked hearing the musicians that play different scales and different intervals, even though that was not part of my original training, I found myself pulling the hearing aid out, just listening with the cochlear implant, pulling that out, then putting both in to see how I could better understand those different intervals. And so here's engagement, not just with the group, but with music and the hearing loss. So really exploring, well, this is how the cochlear implant sound, this is how the hearing aid sound, this is how they sound together. So that kind of interaction. And then for relationship building, an example would be, well, partly because it's a community of similarly afflicted people with similar interests in music, partly because I'm learning a lot about instruments and music that I would enjoy even if my hearing was perfect. And another individual said, this is my connection to others who struggle with music. So these are just like two snapshots, but the results of this, more of this psychosocial understanding of the factors related to the challenges that cochlear implant users face and the solution that they embrace to get past those challenges has been reported in a study with my colleagues in the International Journal of Community Music. And so with that, I want to get into perception. How good is it? How well can somebody with a cochlear implant hear with their cochlear implant? And so I'm going to give you kind of an executive summary, and I'm always a little bit hesitant to do so because, of course, in science, everything is nuanced. In life, everything is nuanced. But in sort of a high level, the musical aspects of beat, rhythm, and tempo are pretty easy for a cochlear implant user. Cochlear implants stimulate with millisecond precision and not microsecond precision, which is something I push for in the lab. But they get down to millisecond precision. So when you're talking about something at beat, rhythm, and tempo, where you're talking about a couple times per second, that's well encoded and tends to be well heard by cochlear implant users. The exception would be if it's as you make the music more dynamic or polyrhythmic or polyinstrumental, then some of the nuances might creep in and might become more difficult to listen for the beat. But in general, that's easy compared to pitch, melody, and texture, which is hard. Pitch is sort of the piano player with mittens on. That's the fundamental problem that percolates up to other aspects of music listening. Harmony and timbre tend to be a mixed bag. With harmony, one of the primary ways that we all hear consonance and dissonance in music is the interaction of the harmonic structure or their partials. And as they get close to each other, they beat against each other. And cochlear implant users can hear that. It's well encoded. So there's an aspect of harmony that is conveyed, and I'll get into the details of that. But there's also an aspect related to the specific location of the partials in terms of their absolute frequency, which is not well conveyed because of the loss of pitch resolution. Timbre, that aspect of music that allows you to hear the difference between a violin and a xylophone, if it's playing the same pitch at the same loudness, that's also a mixed bag. It's a mixed bag because the temporal envelope, the attack and release time of the instrument is well conveyed and well heard by cochlear implant users. But something more subtle, like the roll-off of the partials as a function of frequency, is distorted by that piano player wearing mittens, and so there's some loss there. So it tends to be a little bit mixed. And then tonality, the ability to derive an emotion or a difference in the music based on the key, whether it's a major key or a minor key, that seems to be completely missing. And I'm going to show you some evidence of that. And I see that as a strong motivation to look at tonality with cochlear implant users to try to restore it. Really, it's like a holy grail in the sense that if we can restore that, we pretty much have restored everything. So with pitch, and I want to highlight some of my influences along the way and some of the leaders. Louis Brader was my PhD advisor, and he's done a lot of psychoacoustic works, particularly with hearing aids. And so he was very formative to my thinking on this problem. So with pitch, back to this picture, there's only 22 electrodes trying to stimulate the auditory nerve, and the auditory nerve has 30,000 neurons in it. And so the pitch is the essential problem. The piano player has mittens on. And so with that, okay, there's there's a limit on the technology in terms of how many electrodes. Well, how good is it? How well can somebody with a cochlear implant hear pitch compared to somebody with normal hearing? And so to start things off, I'm showing you normal hearing, frequency discrimination or pitch discrimination for pure tones. And we can look at that with complex tones or piano notes or whatever, but this is pure tones. Here's frequency. This is just noticeable difference over here. So smaller is better. This dashed line right here is one semitone, or the smallest musical interval typically used in Western music, one semitone or a half step right there. And normal hearing, this summary curve was taken from a review article. So this is a meta-analysis over several studies, just showing that normal hearing listeners tend to have, of course, better than a semitone resolution, but actually much better. So being able to tell the difference between 100 and 101 over here, having a sweet spot down like 1,000 over here, you can tell the difference between 1,000 and about 1,002 maybe. And so it's highly resolved. And again, semitones is coming down to about 1/16 of a semitone. These are cochlear implant users measured in my lab and the same type of frequency discrimination. So this is, in my mind, a really good sort of basis for understanding the resolution of a cochlear implant compared to the resolution of normal hearing, and that we can think about how it percolates up to other measures. And so the very first thing I want you to notice, because I'm an optimist, is that a lot of these cochlear implant users are getting into the normal hearing range for sort of the mid frequencies here. And that's pretty impressive. So the other thing I want you to notice is that even the averages are getting less than a semitone here. And so that's essential if you want to be able to hear the difference between a major and a minor mode in music. You need better than a semitone resolution. But then I also want you to notice just a broad range of outcomes with many cochlear implant users requiring 4 semitones or more to be able to hear a pitch difference. And I also want you to see that it tends to be particularly bad for low frequencies over here. And I think that's important for me and the people in my lab, because we're really focused on cochlear implant stimulation timing to provide better resolution. So how is frequency encoded in normal hearing? So in normal hearing, I mentioned that the auditory nerve has 30,000 neurons. There's a place code for that. So different neurons encode different frequencies. So there's a tonotopic or frequency to place coding or place coding of frequency. But there's also the synchronous activity of the auditory nerve. So there's a time code. So I'm just going to show you a couple pictures for that. And so what I'm showing here is the auditory nerve response as modeled by a computational model developed by Laura Carney and many of her colleagues around the world, and then reported by Delaney and all in 2014. But basically, I'm just taking the pure tone and showing you everybody's best guess as to what the auditory nerve is doing at that time. These are the neurons that are firing, so we're modeling different fibers over here. And this is the onset of the tone, just the first 20 milliseconds. And then what I want you to see is when we go from a 500 hertz tone to a 1,000 hertz tone, the place of excitation changes. That's the hallmark of the auditory system, that's tonotopy. But I just wanted to tell everybody and remind everybody that the rate that it responds is faster for the 1,000. Basically, if it's 1,000 hertz, then the auditory nerve is synchronously firing to the 1,000 hertz. If you're tuning your instrument to A440 and you can look at your auditory nerve, the action potentials are phase locked to 440 hertz. So there's a time code. So in normal hearing, you have these two pieces of information that co-vary for frequency. The place and the rate of stimulation or of response. Okay, so what limits pitch resolution in cochlear implant users? Well, one is that piano player wearing mittens, there's only 22 electrodes at most, and it's fewer than one electrode in some devices. So place coding is limited by that number of electrodes, by the neural health, because if the person lost their hearing, there's probably some loss of the vitality of the auditory nerve, and by current spread. So the electrical current is going to come broadly out of these electrodes. And so this is just showing that. So if you have an electrode array here, not only do you only have 22 at most, maybe 16, maybe 12, maybe only one, but the current's going to come out broad and spread before it gets to the nerve. And if you have nearby electrodes, there's going to be a lot of interference, and oftentimes you're exciting the same neural population. So there's really these are the mittens on the piano player. But in addition to that, most cochlear implants, they don't even try to use stimulation timing to convey this level of frequency information. They don't use variable stimulation rate. And so most clinical cochlear implants have a base stimulation rate, for example, 900 hertz. And regardless of what sound that you put in, so if it's a 500 hertz tone or a 1,000 hertz tone, you see the place which electrode is firing is going to change. So it's trying to take advantage of tonotopy, that place cue. But the rate of stimulation is not changing. If it's 500, it stimulates at 900. If it's 1,000, it stimulates at 900. If it's A440, it stimulates at 900. And so it's not using stimulation timing well as a cue. Now, that's not uniformly true. There's one company that is exploring the use of phase-locked stimulation to the incoming sound. That's Mel Corporation. So there are attempts being made at that, and that's something we focus on in my lab. But despite these limitations, many cochlear implant users have pitch resolution that is better than a semitone. So this is really impressive. Shows the power of the brain and neuroplasticity. And so this is where we are. This is the kind of like as good as it gets for today, and we're trying to make it better with better technology, and shrink the size of those mittens, maybe turn them into gloves. So with that, I want to make one more point about pitch before moving on to harmony. In that, the point about pitch that I want to make is that children that are born deaf and implanted after 3 years of age, they tend to struggle more than or somebody implanted at an earlier age on pitch reception tasks. And so I'm going to show you some results from a paper led by Yael Zalt on voice discrimination by adults with cochlear implants, the benefits of early implantation for vocal tract length perception. And the basic idea is we're taking spoken speech, and then we're going to use computer software to manipulate it to either change the voice pitch, we're going to change the vocal tract length, or we're going to change both of those cues. And then we tested 18 adult cochlear implant users. So they're adults. When we tested them, the average age was 25, plus or minus 4. But they were all prelingually deafened. They were all born deaf, and they got their implant at different ages. They all had at least 2 years of experience with their implants. And we measured their sensitivity to those F0 or fundamental frequency differences, or the vocal tract length differences. And they described both of those as being pitch, although some people would say that the vocal tract length is more like timbre because it's like the instrument body. But they both sounded and were described as being pitch-like by the participants. And so what I want you to see here is just the result. This is the age of implantation here. Again, they're all in their late 20s or 30s over here in terms of their chronological age. But this is when they got their cochlear implant. And the basic trend, so this is the DL, or difference alignments, it's the just noticeable difference, so the same thing that I was showing you before. Lower is better. These individuals here can get close to a semitone and hear the F0 difference. And then as we see as the age of implantation, as they get older, it tends to worsen and then kind of like plateau around 4 semitones. So that's the basic trend. There are outliers, and as you can imagine, it's really difficult when you're working with an adult who's now 30, to be certain that they had no experience with hearing in their first couple years of life. And so these outliers are fascinating to me because they're either they had early formative experience with hearing, and that's why they're outliers, or they actually overcame that deficit and were able to achieve good resolution otherwise. And so trying to figure out why these outliers are outliers is important. But the basic trend is that the later they're implanted, the worse the pitch outcomes are, and even though they've had many years of experience with the cochlear implant now. And the same is true for vocal tract lengths. The basic trend is the later they're implanted, the worse off they are in terms of vocal tract length perception in the combined use of the two cues. And so now I want to talk about harmony. And so for harmony, and I think I got into harmony because it's one of the things that's really misunderstood in the cochlear implant field. I often hear that, cochlear implant users can't hear harmony. And I'm like, that doesn't sound right. I feel like I can hear harmony. So let's take a look at that. And I find that many cochlear implant users can hear harmony really well, and they're in a way that's quite like their normal hearing peers. And this was reported in a study from my lab, pleasantness ratings of musical dyads in cochlear implant users. And this was inspired by a study by Emily Spitzer and colleagues. And so if you take two musical notes and you play them together, I hope I can play them. If you take two musical notes and you play them together, they're either going to sound good or bad together, or more appropriately, they're going to be consonant or dissonant. So if you take a note and you play an octave and you layer them on top of each other, then it sounds smooth and consonant like that. And that sounds smooth and consonant to me. Well, if you take one that's one semitone off, then it sounds dissonant. And if you ask normal hearing listeners to, let me play that again. If you ask normal hearing listeners to rate the consonance or dissonance of this, then, and this has been well established since Pythagoras. He was the first to outline this. But let me get rid of that. The basic trend is that, of course, the octave is consonant, as is a note on top of itself, or unison. Major third and perfect fourth. What that part of this is perfect fourth, tritone, perfect fifth. This is a basic pattern of harmony rating. Now, if we do the same thing with cochlear implant users, we get something similar, but it looks squashed and watered down a little bit. But some of the key features pop out. So you see, of course, unison and the octave. You see the perfect fourth and tritone and perfect fifth making a little dip like that. So some of the features are coming out, but it seems like squashed and muted a little bit. Now, if you're testing a group of people and you see something like this, you got to think like, well, amongst this group, there's got to be people who are really good at this, or they have like normal patterning, whereas there's some that have more abnormal patterning. And so we wanted to look at that. So basically, to look at that, we're going to look at all of the individuals' ratings against this normal hearing kind of prototypical curve to see how the ratings differ amongst cochlear implant users. And when we do that, we see that the normal hearing listeners are all very self-consistent. They kind of agree with each other for the most part. So this is the correlation. So how well correlated their ratings were to the normal hearing average. And when you look at the CI users, again, they're all over the place. The first thing that excites me the most, actually it all excites me, but the thing that excites me perhaps the most is that many of them have near normal harmony perception. And if getting into the nuts and bolts of why, it makes sense because, again, part of harmony is caused by the beating of the partials when they're just off by a little bit. And that appears in what's referred to as the temporal envelope in cochlear implants. But there's also many CI users who have actually no correlation or anti-correlation. And so this excites me too, because I know, these guys can do it, and I know the signal processing, so I know that they're getting the information. So a lot of the efforts in my lab is, well, can we train this? Can we improve how well these individuals can hear harmony through training programs, kind of like reintroducing them to it? Okay, and then finally, I want to end on tonality. And so with tonality, the point that I'm trying to make is that CI users, they can't hear tonality well at all. I think it's completely absent. I'm going to show you why I think it's completely absent from one of the studies that we did in the lab, and then I'm going to talk a little bit about how we're going to try to approach it in the future. And this was inspired by a study by Amber Dahan and colleagues. They were looking at some similar concerns. And so by tonality, I mean that really the difference between major and minor modes of music, or major and minor scales, which is just a semitone difference between the scales. The third, sixth, and seventh note are lowered by a semitone or a half step. And changing a melody from one of these keys to another produces quite a large emotional response in people with normal hearing. And so I'm told, I can't hear it myself, and that's what we're going to get into. And so in this, the study that we did, we took melodies that were originally peaceful, and we converted them to, all the peaceful melodies were written in a major mode, and we converted them to a minor mode. And then conversely, we did the same thing with sad, which were all written in a minor mode, and we switched them to major. But before we did that, we wanted to do a validation study, because that earlier study by Amber Dahan and colleagues, they were looking at this with happy, sad, scary, and peaceful melodies. A little short excerpt. I'll give you an example of one in a second. And they had people with no known hearing loss and cochlear implant users rate the valence, whether it's pleasant or unpleasant, and the arousal, how relaxing or stimulating it was. And then the same thing for CI users. So this is the data from my lab. We kind of replicated this part. And then, there's nothing really surprising here. If it's happy, it's stimulating and pleasant. If it's peaceful, it's more relaxing but still pleasant. And then sad becomes more unpleasant and but still relaxing. And then stimulating and unpleasant would be the scary ones. Again, the cochlear implant users are much more squashed and compressed, and there seems to be less differentiation, except for the happy ones, which tends to be really at a upbeat tempo and have more frills to it, so they could hear that kind of out. But I was really interested in this difference right here between peaceful melodies and the sad melodies. Because if you just look at these results, you might think, well, maybe they can hear the difference between major and minor. But we took a really close look at these original melodies used by this other study. And in the other study, there was a tempo difference. So you could see that the sad songs tended to be a little bit less than 60 beats per minute, and the peaceful songs to be a little bit above 60 beats per minute. So it's quite possible that what was driving the valence ratings for the cochlear implant users was just this tempo difference. So we synthesized all of these melodies, or just the peaceful and the sad melodies, to be exactly 60 beats per minute. And then we retested everybody, and then we switched from major to minor mode and from minor to major mode. And let's skip over that. And when we did that, and so let me start off by playing. So this was an originally sad song that was done at 60 beats per minute. And this was modified from minor to major. Now, I have no idea how big of a difference that is for you. But, you know, when we tested the normal hearing listeners, we made that switch. The sad songs, when we switched it from minor to major, they jumped up here into the valence. And then the peaceful songs, when we switched it from major to minor, it dropped down here. And so there's a big switch. Where with the cochlear implant users, squashed together. And then all the data of this paper, we're analyzing it in different ways. There's no evidence that the CI users can hear this change. And I'm kind of ending the data on this point, even though, you know, I'm an optimist, but I kind of like having this as a litmus test. It's kind of like, oh, this is something I can't do. And even the star CI users, some of them are like gigging jazz piano players and piano teachers, and they have a lot of music experience, and they're not getting this key aspect. And so even though that's kind of this is something that's completely missing, how do we get at that? I want to end on a positive note, which I tongue-in-cheek call the first law of cochlear implants, but it's really true. It's that the more you listen, the better you hear. And so in my lab, we're doing a lot of training studies. How can we give harmony back? How can we give tonality back? And we also do a lot of psychoacoustic studies of digging into the details of that. Can, for example, in Peter's talk, the use of cadences, I think could go a long way to understanding how well some of that tonality differences affect listening. So we're doing a lot of training studies. And so with that, just to wrap up, the main point is that music appreciation, whether or not somebody likes music, is really what you make out of it. So we're diving into the psychological and sociological factors that affect outcomes and understanding the challenges faced and the solution embraced by cochlear implant users. Pitch perception widely varies amongst cochlear implant users, but resolution can be better than a semitone. And my lab is working through both rehabilitation and better technology to keep trying to move into that normal hearing range. Many cochlear implant users hear aspects of harmony just like their peers with normal hearing, although there's a lot of variability there, and we're trying to bring those that struggle up. And then tonality perception is particularly challenging since it requires pitch resolution better than a semitone. But not only that, it requires the building up of these associations between the music and the emotion. One other last point I'd like to make on that is that most of the CI users that I work with are postlingually deafened. And so I want to start exploring this in prelingually born deaf, early implanted people. And then acknowledgement. My work is funded by 2 project grants. One, NIDCD, Encoding Temporal Fine Structure for Cochlear Implants. What I didn't talk about today is more the signal processing side of things. And then a project grant on music appreciation after cochlear implantation, which I did talk about today. And then a really nice training program in the Hearing Communication Neuroscience program, which we're celebrating today. And on that note, I wanted to say that, you know, I've had 3 PhD students and a postdoc supported by this training program. And if anybody wants to know anything more about it, feel free to contact me or, of course, Chris Shera. Thank you.
You can make 1, 2 questions. 1 or 2 questions.
Hi, Ray. Thank you for the talk. I had a couple questions about the study where you looked at F0 and vocal tract length. The first is you talk about manipulating vocal tract length in the sentences. How did you do that? What do you mean by that? And second, in that study, how do you think about disentangling, F0 and vocal tract length? Because to me, it seems like, there's a relation between these two. Like a larger vocal tract has like typically a lower F0 So there's some correlation between these two aspects. So, yeah.
Yeah, I'm going to have to ask somebody to repeat that question. Maybe Asar, if you could come up. It's just the room acoustics are a little bit difficult.
I think your question was about how did he manipulate the vocal track?
How are we controlling for the vocal tract?
No, in the experiment that you did, how did you manipulate the vocal tract?
Yeah, and so I was not the one actually controlling, doing the manipulations, but we were using some kind of synthesizer, like Clat synthesizer or straight. I think it might have been straight. And so, however, the straight software controls for vocal track is how we did it. But in terms of what we did, we changed the length to have semitone differences in the scale. At really small differences, and then we put together a discrimination task to say is it higher or lower. So they're basically having to say the subject task was it higher or lower, and then we adaptively brought it down to see what their just noticeable difference was.
Did I repeat your question completely? So disentangling the vocal track size from the F0 frequency.
Yeah, I that was not my contribution to that study.
Great. Okay, so with that answer, let's go to lunch.
Yeah.
Let's thank Ray. We're back. Did everybody enjoy lunch? We were lucky with a beautiful day. So now we're going to have a musical performance by one of my favorite bands, Delirium Musicum. They were gracious enough to, generally they're a larger band, but just a section of them are here today. Etienne and his group have been friends of the Brain and Creativity Institute for a long time. Etienne did his music studies here at USC. So without further ado, let's welcome Delirium Musicum.
afternoon, everyone. Sleepy after so long. It's very nice to be a little bit part of this day that you have here, and I'm looking forward to listening what you will have to share just after we play. But you probably recognize a Brahms Hungarian dance before. The reason we played that is because I have this wonderful orchestra called Delirium Musicum, which is about 15 musicians. It's a self-conducted string orchestra. And we're actually playing at Disney Hall on November 16th at 1 p.m.
Some of them have been to our concert here.
Oh yeah, we played here many, many times. Anyone has heard Delirium Musicum here? Well, if you haven't heard, and if you have heard us here, you can also hear us on November 16 at 1 p.m. at Disney Hall, and you can find also information about that on the website, deliriummusicum.com. The reason we're only two now is because during the pandemic, we could not play with a whole band, so we created what's called Musikcaravan, which was Yoon and I and Boris the boss traveling for 6 months between the Mexican and the Canadian border. Boris the bus is a 1971 VW bus.
50 years old.
50 plus years old, with his own personality. I'll let you imagine what that means. And if you cannot imagine it, although I'm sure you can very well, you can see it with your own eyes online on YouTube or on the website of musikcaravan.com, because we have 15 episodes filmed from that journey, from us playing for ostriches, for turkeys, even for some humans, whether they liked it or not. Oh yeah, Musikcaravan with a K, just in case you're good at spelling. For the next piece, we're going to play a piece that felt a little bit like how we felt during the Musikcaravan tour, because before the pandemic, either Yoon was traveling when I was in Los Angeles, or I was traveling when she was home. So we basically never seen each other. Mm-hmm. And during the pandemic, that changed when we lived in 4 square feet for 6 months. So the next piece is by Prokofiev, and he's writing about the revolution, and you have all the gunshots and so on. In his sonata for 2 violins, this is a second movement. But although sometimes we felt like this during Musikcaravan, living together in that small space of Boris the Bus, there's always sound after the rain. And our song today will be Javier Iglesias Martin, our favorite cellist from Delirium Musicum. So if you'd like to join us and show some love for Javi. Do some words. So we're going to play some Shostakovich for you, but don't worry, this is not the angry, heavy Shostakovich of later on his symphonies and quartets. This is a very young Shostakovich, someone who is still young and innocent, just like we were before Musikcaravan. This is from his Jazz Suite number 2. You might know it from New Year's concerts, from the Vienna Philharmonic and so on. And this is a waltz. So if you feel like dancing or grabbing the arm of whoever is next to you, go for it.
Say about 5 to 7 minutes.
Oh. Then we're gonna play the last piece for you before we get back to like more serious stuff. Try that.
What does that mean?
Oh. So we're going to play not a Hungarian piece, but a piece by an Italian composer who wrote in the Hungarian style. The name is Vittorio Monti, and the name of the piece, the title of the piece is Csárdas, which you might also have heard. And this is also one of the pieces we played a lot during Music Caravan in any wintry, snowy, or summery conditions. And Javier here is making it just so much better by adding this third dimension to it.
social media.
Find us on social media if you guys have some videos, some photos, and tell us hi, and we'll answer you. Hope to see you guys soon. Thank you.
So, Bob, not a hard act to follow. Etienne one time invited me to give a concert, to a talk as part of the series baby concerts that they give. They provide this beautiful music to parents and their young children. And that was the toughest talk I've ever given because there were a bunch of 2-year-olds in front of me while I was trying to talk to the parents. And at some point was like, no, this is, this is much nicer just to watch. Okay, so we are continuing. We have 3 more talks for the afternoon. Our first one is going to be by Bob Slevc, Associate Professor and Director of Graduate Studies at University of Maryland. Bob is the Associate Chair in the Department of Psychology, as well as part of the program in Neuroscience and Cognitive Science and the Maryland Language Science Center. He received his PhD at the University of California, San Diego, and was a postdoctoral scholar at Rice University before going to UMD. His research focuses on the cognitive mechanisms involved in the processing of language and of music in both normal and brain-damaged populations. Let's welcome Bob.
Yeah, I guess I'll put it there. Cool. Oops. Okay. I didn't bring my violin, so I'll just have to talk. So music and language. Why would I want to talk about music and language besides that I was invited to do so? I mean, I think people have been interested in music and language for a long time, and I think one of the reasons is that they seem to have some kinds of similarities, and also that we don't really have good animal models, for example, for either one, right? They both seem to be these kind of human-specific skills. They seem to be universal to humans. Every normally developing person develops this kind of impressive ability to use and understand their own native language system, their own cultural musical system. And we just don't have great evidence of this in anywhere near that kind of productive way in non-human species. And so I think the hope is that you can get some insight into how language works and into how music works by trying to understand how they might be related. Now I'm saying music and language, but what exactly are we talking about here? I think we can all agree that what we just heard was music. Hilary Hahn here playing the violin, I think, is music. And over there's Ben Stein and Ferris Bueller's Day Off, which probably a lot of you are too young for, but sort of monotone speaking economics professor, we can argue is language, right? But of course, there's like a whole continuum of things in between, right? So you have Bob Dylan won the Nobel Prize for Literature for some reason. You've got rap, that's Nas. You've got Amanda Gorman, the poet, or like beat poetry, or you have like coordinated chanting, at rallies and things like that. And it's sort of like, how do you draw the line if you want to try to compare these things? Now, fortunately, there's a bunch of us who have been working on music and language for a long time, and so I think you can kind of look at how we've solved this problem, which is basically we've decided that music is Bach and language is Chomsky's generative grammar. Sometimes Mozart, but that's about it. Obviously, this gets even worse when you start thinking about like different cultures, different cultural musical systems, different language systems, and I think I just want to sort of put this up front and say, I think as a field, as my little subfield, we've done a very bad job sort of thinking about it beyond this. But, and I say this partially because I think these sort of implicit prototypes of what music and language are have really influenced the kinds of questions we ask. And so a lot of us got started out by noticing that like, hey, a really interesting parallel between music and language, at least defined this way, is that they both have this sort of rich structure, right? So in language, you have these sort of hierarchical structures in which discrete linguistic elements can get combined into these sort of infinitely generative sequences by the rules of generative syntax or something like that. And similarly in music, you can think about combining discrete musical elements into sort of complex, generative, infinitely complex sequences according to the principles of some kind of musical structure. And so this raises this question of whether they might rely on the same kind of shared cognitive and neural systems, right? Maybe we can get some insight into this by doing cross domain comparisons. To be fair, this is the comparison that we like to make. The comparison that most of us have actually made is more like this, which is you can talk about hierarchy and hierarchical structure in music in a couple of ways, right? You could talk about the sort of structure and the relationship of elements in a sequence, which is what I'm doing here, but you could also think about the sort of relative hierarchical importance of pitches here in a tonal context, right? And that's another kind of hierarchy. And so a lot of the work is really focusing more on this kind of thing. What this is, by the way, is just a multidimensional scaling solution of the relative stability or of pitches in a tonal context. So it's oriented towards C. At the bottom, maybe I can use this thing. Maybe. Whoa. I'm not going to do that. You can see that like C and C prime, the octave above, are very closely related. They're relatively distantly related to the things sort of up on the top. And this is all based on some really nice work by Carol Krumhansel a long time ago. In any case, for our purposes, it doesn't totally matter, except to say that a bunch of us have wondered if these might rely on the same kind of cognitive and neural systems. One way that this has been addressed, right, is by talking about sort of neural overlap, right? So there's lots of evidence that aspects of musical structure seem to implicate areas that are often associated with linguistic structure or linguistic syntax. This is not a real study. This is me pulling peak activations from the Neuroscience database for about 100 studies of syntax in fMRI and about 100 studies of music in fMRI and just overlaying the activations, the peak activation. So this is not in the same people or anything like this, and this should be taken with like large dashes of salt. But one of the things you'll notice is there's a fair amount of overlap in green, right? And a lot of that's temporal lobes, like you might expect, but there's also a lot of like left frontal stuff, which is kind of a classic like language syntaxy sort of area. So that's kind of interesting. On the other hand, we've known for a long time that there are dissociations in language and musical deficits following brain damage, right? So most relevant to structure, which is what I'm talking about so far, we know that there are people who appear to have preserved syntactic processing in language despite harmonic processing deficits, right? So you can have, there's a very like carefully tested case from Isabelle Peretz a long time ago now of a patient who had bilateral aneurysms and really lost basically all sensitivity to tonal structure in music, but was apparently totally fine in terms of syntactic structure. So that's sort of a problem if you think there's some kind of one-to-one correspondence. Now, in neuropsych, we always want double dissociations because you could just say, well, maybe, you know, like the language structure was more well-learned or something. But a few years ago, we came across a patient who showed the opposite pattern. So this is a post-stroke patient who was very severely agrammatic in language, but still had normal sensitivity to harmonic structure in every kind of way that we could test it. So, you know, that seems to suggest that these are pretty separable, at least at a sort of neural implementation kind of level. And so how might you reconcile these kinds of findings? One influential approach comes from some of Ani Patel's work, where he proposed that what you might actually have are just shared syntactic processes, but separate representations, right? So that you might have some shared kind of computational processes that act over musical representations or that act over linguistic representations. And so those representations can be selectively damaged, but the overlap reflects sort of shared processes on those representations, right? The prediction you can make from that is that if you have simultaneous demands on those, you might expect them to interfere, right? If I'm having to do a lot of hard structural work in music and a lot of hard structural work in language at the same time, you know, that might be difficult. And there's various bits of evidence that support that. So it turns out that manipulations of harmonic structure can interact with processing of syntactic violations in language, can interact with the processing of complex syntax, and in the processing and resolution of syntactic ambiguity in language. And I want to talk a little bit about that last point for obvious reasons. This is actually the study that got me like really into music psychology. So this is sort of like, for me, this is like old history. I will just say as an aside, this talk is going to be a little bit of a tour of like things Bob has been wrong about. But I think that's okay. I think we learn things from being wrong. Okay, so what we did in this is we took Ani was involved in this project, actually. We took Ani's theory here, Ani Patel's theory, and said, well, let's see what happens if we make people try to resolve temporary syntactic ambiguities, which I'm going to call linguistic garden paths. These are really well studied in psycholinguistics. And we relied on this particular kind of ambiguity where you can have something like the scientist confirm the hypothesis. The hypothesis here can play a couple of roles at this point in the sentence, right? You're processing incrementally, you hear things as they come along or read things as they come along, and the hypothesis could be the direct object, the thing that was confirmed. Right, like confirmed the hypothesis yesterday. But the hypothesis also could be the subject of an embedded clause, right? So maybe the scientist confirmed the hypothesis was false, right? And now the scientist has confirmed something, which is a whole other sentence thing about the hypothesis being false. And you can see, like a sort of nice comparison here is just by inserting the complementizer that. If I say the scientist confirmed that the hypothesis, now I can't say yesterday, right? Now it's unambiguously a sentence complement. And so you can compare these conditions. This has been done a lot. And you can show that if you do something like word-by-word reading, where I flash words up on the screen and you have to push a button for the next one, people are slower to read the verb in the ambiguous condition on the top, where you probably have to now sort of say like, oh, the hypothesis isn't the thing that was proved, it's something about the hypothesis, versus in the bottom where you knew there was going to be a verb coming. Okay? That's not that interesting. I mean, it is interesting. Lots of people have done this stuff, lots of arguments about exactly what's going on there. But whatever's going on there, it's something syntactic, is the story. So we did that, and at the same time, we played chords, right? And so it's a pretty simple task. We're doing the same syntactic ambiguity manipulation. We're looking at how much syntactic ambiguity influences people, but we're doing it when you simultaneously get a chord that you either expect harmonically or that you don't, right? So just to give you a sense, this is sort of what it looked like. I'm gonna be the participant. We'll see if this works. So people do that, and we measure how long it takes them. So what I'm plotting here is the difference between the ambiguous and the unambiguous condition. And basically what that means is above 0 is how much slower they were when it was an ambiguous condition without the that than when it was a non-ambiguous condition with the that. And so you see a garden path effect where people were slower to read the verb when it was ambiguous. That's not surprising. This is paired with in-key chords. When we had the out-of-key chord at the same time, the effect was much bigger, right? So this is exactly the kind of interaction we expected. I was so excited. I mean, I was like, this is amazing. Getting out of the psycholinguistics business altogether. Now, you might say like, well, maybe it just was distracting, right? It sounded weird. Maybe people just stopped paying attention, slows you down. So we had some controls. One is we tried a kind of semantic ambiguity, where we said, well, or not ambiguity, semantic unexpectancy. So we said, if you read something like the boss warned the mailman to watch for angry pigs, people are kind of slow compared to dogs when delivering the mail. And so we did the exact same thing. We find, again, this is the difference between the expected and unexpected condition. We find a semantic unexpectancy effect, but it doesn't interact with the harmonic manipulation, right? So we say, okay, it seems to be something specific to syntax. We're like on firm ground. But, maybe you think, well, like, I don't know, maybe this dogs, pigs thing is like not that hard or something. So we tried another control. Oh, well, so here's what we found, right? We get this interaction between language syntax and harmonic, simultaneous harmonic manipulations, but not between semantic manipulation, language, and harmonic manipulations. As another control, we decided, well, we'll keep the same sequence, but we'll manipulate the timbre so that it's like unexpected, but not structurally unexpected, right? There's nothing about the sort of hierarchical structure that's unexpected just because it's played by a different instrument all of a sudden. It went kind of like this. So it's like, it's distracting. Right? You might think that if it's just sort of being distracting or kind of weird, that ought to capture your attention. But in fact, we found no interaction for syntactic or semantic violations in language. So again, I was like really excited. I was like, this is so great. I'm even more excited when a couple of people from Psyche's lab were able to basically replicate this effect because, holy crap, I was always sort of afraid to try to replicate it. But here's the thing. You might look at this and you might say like, well, that's sort of a good control, but maybe that's not that good of a control. Because in the first case, when I'm calling the syntactic one, you also have to like reinterpret something that happened before, right? In the second case, you just have to kind of deal with something that's a little bit unexpected. So maybe it's not just a structure, like meaning kind of difference. What you really might want is something like what you might call a semantic garden path. Like the old man went to the bank to withdraw his net instead of withdraw his money, right? Where you're like, oh, now I have to realize that bank meant like meant like side of river, not financial institution, right? Not structural, right? It's still playing the exact same role syntactically, but it requires you sort of reinterpreting something. This is not my idea. This is Petr and Petr Janata did this in French, but they get a pattern that looks a lot like the pattern that we got, but now not with syntax, right? Just for comparison purposes, there's my data. The pattern looks pretty similar. So what does that mean, right? I mean, something's shared. You're getting this interesting pattern of you're getting interactive effects in some cases and not in other cases, but it doesn't seem to be tracking with syntax or structure exactly, right? Now, around this time, I was talking with a colleague of mine at Maryland, Jared Novick, and Jared had a whole theory of garden path sentences and sort of syntactic ambiguity in language, where he said, oh, it's not that you have some syntactic processor, right? What you have is domain general mechanisms of conflict resolution, where you have a conflict between the sort of interpretation you're building and this incoming information, and you have to detect and resolve that conflict. And he basically says this is classic cognitive control. It relies on the left inferior frontal regions, right? These are regions that have often been implicated in music, language, sort of neuroimaging studies. So like maybe what's going on is that it's not that there's shared syntactic processes, it's just that these things both have this particular kind of complexity that requires cognitive control to kind of come in and adjudicate in these situations. Now, if that's true, then one thing that we should be able to do is to make sort of musical garden paths, right? There should be these points in music where you kind of have some interpretation and you have to like reinterpret something in the past. I'm going to try something. What I'd like you to do is just tap along to this music, okay? But like, what happens, right? What happens is you have this really long syncopated opening, and the drums really strongly project this kind of triple meter, right? And then as more instruments and singing stuff comes in, you sort of realize it's just a very heavily syncopated, 6/8 or something like that. And that's the kind of thing that, that we think might be like a kind of musical garden path, right? You have an interpretation of something, you've kind of determined a meter, now you have to kind of reinterpret that. That said, I was just talking to Jessica about whether she sees any left frontal activation in this sort of similar kind of construct in the Cameron and Grahn paper, and they don't. So
we don't kick people out. It might be different.
It might be different. Yeah, there's hope. But this is probably another case of Bob being wrong, but that's okay. There's various other kinds of cases of this. My student Brooke Okada a few years ago and I worked out a bunch of different kinds of examples of things where you might imagine this, like one in back into harmonics, you can think about pivot chords when you change keys, right, where you have a chord that plays a particular harmonic function, and then as it goes on, you sort of have to realize that it's actually playing two harmonic functions at the same time. You could think about counterpoint, right? You think about sort of contrapuntal ambiguities where you have a line that you think is maybe two parts, and then you get some counterpoint thing that suggests it's actually one kind of stream, something like that. So plausibly, those might be what we're talking about. What does that mean? Well, I started by saying we might be able to figure out something about the shared syntactic processing in language and music, but I think, something's shared, but it doesn't seem like it's syntax, at least from these studies, right? Maybe it's something more general, like shared reliance on cognitive control or working memory or other kinds of sort of more basic cognitive processes, right? Another possibility is that we shouldn't be talking about syntax at all, right? And it's really just this sort of way that we framed our question as being sort of a Bach versus Chomsky kind of story. But really, maybe we ought to be talking about, prosodic structure in music, right? Prosody seems a little more closely related to music anyway. Prosodic structure is hierarchical, right? But it has some ways in which it seems to be different than sort of syntax in language. And maybe that's the sort of parallels we really ought to be looking for. Now, going from music and prosody, you might say like, well, yeah, actually, like it's funny that we started with structure anyway. Like, what a strange place to start when looking for music-language relationships. We ought to be talking about sound, right? Music and language have these sort of temporally unfolding sequences of sound. You have to interpret them. You have to work them into a context, And so you might think like, well, maybe we should just even very strongly modular theories of music or language are going to argue that, like, of course, the early stages of music and speech processing have got to be the same, right? You're just like at some point, you're just recognizing auditory objects and stuff like that. So maybe we can kind of look at how far up the sort of sound processing stream we can go how before you get divergences, right? Indeed, there's a lot of evidence over the years that aspects of musicality predict aspects of speech processing. As an aside, another thing I think we don't have a good handle on in this world is how you define musicality, right? What counts as being a musician? Do you need to be a musician to count as somebody who is musically savvy, right? These are all, I think, good questions. But in any case, different aspects of musicality predict aspects of speech processing. That includes things like speech and noise processing. So musicians or people who are better on musical tasks tend to be better at understanding speech and noise. There's a nice meta-analysis on this from Assel's group. Phonological awareness seems to be related to musical ability, sophistication, musicality, right? And phonological awareness is really critical for learning to read and further kind of processes like that. And so that can explain some of those kinds of relationships that you see. Sensitivity to prosody. These are all meta-analyses, by the way, so there's enough data out there that you can feel, I think, pretty confident about it. And in some work that I've done over the years, and actually that my student, Rachel Thompson, has just recently done, we find that musicality, sort of broadly speaking, relates to adults' ability to sort of successfully learn the prosody of a second language, or the pronunciation of a second, not the prosody, the phonology of a second language, right? That's a classically hard problem in second language acquisition, kind of being able to speak with a more native-like accent to understand contrasts that don't exist in your own language and so forth. For those of you who aren't familiar with meta-analyses, this is a very impossible to see figure, but it doesn't matter, because all you want to see is that most of those dots are to the right of that dotted line, right? These are all studies, and these are their effects, right? How much better musicians or music-y people were than non-music-y people. And you see that this little tiny dot way on the bottom is the overall meta-analytic effect. So okay, maybe the sound processing is pretty shared. That would explain these kinds of relationships. If it's hey, being good at music is being good at the same kinds of sound processing that you're using in language. But on the other hand, there is other kinds of evidence that separate neural resources appear to be attuned to particular classes of sounds like music and language. So this is one example. This is an example I like, where basically what they did is they got people to listen in the MRI to, this is not me, but listen to a whole bunch of different sounds, and then they did some sort of principal components analyses on the neural data, where they sort of say, well, what are the kind of properties that you might be able to extract that separate these different, that categorize these sounds? And a lot of them are things that you expect, like sensitivity for frequency and spectrotemporal modulation and stuff. But two of them, the ones on the end here, seem to be these components that are really specific to language or to speech and to music. And speech includes, to some extent, music with vocals and stuff. But you see, basically up here, high things are things that factor cares about a lot, or that component cares about a lot. Things near the line are things it doesn't care about. And you get these distinct components that seem to suggest sort of separable specialization for musical and linguistic sounds in auditory cortex. And so, the story there is well, that's useful because you want to be able to separate stuff into sort of speechy stuff and music stuff so you can process it separate, appropriately. But that implies that they're processed pretty separately. That said, right, this still relies on a sort of default assumption of what music and language are, right? They're classifying these things by saying, well, okay, I'm going to call whistling music maybe telephone dialing because it's pitchy, and I'm going to call these things speech. But this is sort of top down, and to the extent that we've classified them because they're different, maybe that's not so surprising. So another one of my students, Matt Sanog, and I decided to do what we thought might be a slightly more direct test, which is to look at timbre. Instrumental timbre, right? So what distinguishes, one kind of instrument from another, and voices. So why I sound different than Peter. That's the only difference. Yeah. And so, this isn't crazy. When you look at studies of timbre perception or of voice perception, you see sort of a roughly similar kind of pattern of neural activity associated with. They're both associated with, anterior temporal lobes and planum temporale and stuff. But you can't really interpret overlap between different studies. I mean, I did earlier, but let's ignore that. And so what Matson and I decided to do was to use neuroimaging and machine learning techniques to try to ask if the same bits of brain seem to care about timbral and voice perception, okay? And when I say decoding, basically what you do is you just take, Jessica mentioned this a little bit earlier, but in case anybody wasn't there or that you forgot or whatever, you take patterns of neural data, and you use that to train just a simple machine learning classifier, which is basically a correlation machine. And then you give it held out data, and you see how accurately it can classify what the stimulus was that produced that based on the neural data that it came from, right? Because MRIs are loud, we used a sort of a sequence where we sort of turned off the data collection for a minute so it was quiet, played the sounds, and then turned it back on to collect a few volumes of data. This works because fMRI is a pretty slow response. So even though we don't get data right when they're hearing the sounds, we can pick up the sort of consequences of that processing a little bit later. So the way it worked was sort of like this. We had these different speakers producing little vowel sounds, and we had different instruments producing different pitches. We matched them, so they're lower register instruments to match some of the lower register pitch sounds. It sounds sort of like this. So we can have people listen to a lot of things like that. We train classifiers. We do a kind of searchlight where we go through the brain and we say which parts of the brain seem to care about these kinds of differences, right? Then we do the same thing with the instruments. Right, and we say, okay, what parts care about that? And then we can just do a really stupid thing where we just put those maps on top of each other and we say, is it any of the same bits that seem to care about both of these things? Right, and we're like, well, this is like a first pass, but it'll give us some evidence of like what parts of the brain are involved in or seem to carry information about both musical timbre and speaker voices. So what I'm showing you here, oh, there it is, are the regions that can distinguish. In blue, you can see the bits that can distinguish instruments from each other. And in red, you can see the bits that seem to distinguish speakers from each other. And in kind of an ugly pink, you can see the overlap, except you can't because there isn't any, which was surprising to me. In fact, there was one single searchlight location that could decode both speakers and instruments above chance, but we were expecting more. Now you get a slightly larger region of cortex involved if you just say what can distinguish speakers from timbres, and that's like a lot. Those are maybe more distinct in some ways. But these seem like there's some pretty early specialization going on even at this level of timbral and vowel perception and speaker perception. So what's this based on? To see if it was just about sort of like the acoustic kind of features of the stimuli that let you sort of separate them, we did a simple acoustic localizer. These are just like some simple synthesized tone patterns and amplitude modulated noise and stuff that can kind of pick up a lot of like early auditory processing areas. And that's here in green. And then we can sort of say, well, to what extent are the things that are distinguishing speakers and timbres just part of like earlier auditory processing? And the answer is a lot of it. There we go, a lot of it. So basically all of the instrument and speaker specific things that can distinguish speakers from speakers and instruments from instruments are within this kind of general auditory processing, early auditory processing sensitivity area. That is another, like individual sound sources seem to be represented in separate regions of the temporal cortex, but still regions that are implicated in more general acoustic processes. So where does that leave us? Well, and I should note that fMRI is a fairly slow thing, and so you might think like, well, maybe there's like subtler differences early on in processing. I'm not going to talk about this in really any detail, but we've also done some sort of similar things using MEG. MEG is temporally very good, has very high temporal resolution. And so we can do the same kind of thing where we like record MEG while people listen to a whole bunch of different kinds of sounds. We can train a machine learning classifier to classify. In this case, it's a whole bunch of different environmental sounds, instrument sounds, and speech sounds. And we can just basically tell it, train it on distinguishing each pairwise pair from each other. And so what that matrix is what's called a RDM, response dissimilarity matrix, which is like how different the neural response is for any two pairs of stimuli across all of the stimuli we have. There it looks like a bit of a mess. But you can plot that, and then you can look at how that evolves over time. And the take-home message here, like all I really want to focus on is that sound tokens can be decoded from brain activity after about 90 milliseconds. We have enough data in there just from, that we can already tell, individual sound tokens from each other, especially if they're between category. Right, so you sort of see the emergence of structure where you start to see the three squares of, environment, environment. I'm used to lower screens. Okay, so they can be decoded quickly from each other. It seems like the brain is very quickly sort of discriminating auditory objects from each other, but they seem to implicate mostly separate neural regions for speakers and instruments. These are data from that same MRI study, but this is just the simple univariate contrast where we just take areas where speech processing is greater than instrument and overlap. And you see the overlap in pink, but again, you don't really. So that was surprising. We have these correlations between musical and linguistic abilities. They're pretty robust, but it doesn't seem like those reflect kind of same neural mechanisms like very far up the pathway, right? And so what I would probably argue is that this, again, like the structural thing, might just reflect shared reliance on more kind of basic underlying processes that are important for both language and speech, right? Maybe it's perceptual acuity. Maybe being good at them just means you have to be good at hearing. Maybe it's like auditory category learning processes or something like that. We've been trying to work on that and haven't made a ton of progress yet, but. Okay, well, crap. So where do we go next? I mean, you might think that like, when people think about what music and language do and the human specificity of it and stuff like that, a lot of times what people think about is like, music is, it's meaningful. It's like, it communicates things, it conveys emotions, stuff like that. And so you might say like, well, what about meaning? Certainly music and language can both express emotion in one way or another. So I'm going to play a couple sentences for you and you can sort of think about what emotion they'll be. They're in a language that I think probably most of you will not speak, but I think you can understand the emotional content anyway to some extent. So any votes on what the first one is? Sad, happy, angry? Excited, something like that. Second one? Sad, yeah. And music can do the same thing, right? I can play music for you that'll have different kinds of effective consequences, even if it's a song you know well that might have, I'm going to play you an example. Well, I'll just play it. I'll tell you about it. So what's this? This is, of course, happy. This is Happy Birthday. It's a classic happy song, and you have lots of connotations to tell you it's happy, right? But of course, it sounds like it's horribly depressing. And so that's sort of interesting, right? Like it seems like music and language can communicate these things. Just out of curiosity, did you get it?
Yes.
Yeah, because it has lots of cues, right?
You're very primarily driven by tempo.
Tempo, yeah. I think that's probably a stronger cue a lot of the time. So you might ask if emotional prosody and emotional music are related in some kind of deep way, right? And shout out to Leonard Bernstein. Leonard Bernstein had this whole bit where he liked to refer to the minor third as a musical linguistic universal. This is many of you will know about this, but many of you may not. Bernstein gave this sort of amazing 7-lecture series at Harvard in the 70s, all about music-language relationship. And he sort of is the guy who came up with all the ideas that those of us who study this just are still milking today. But this is one of them. He says, well, the minor third is this musical linguistic universal. It's like a way that you can express sadness in language and in speech and in music, and it's sort of everywhere. There's some data that actually supports that. So several years ago, Megan Curtis did this study where she got actresses and she gave them these just very neutral things like okay, and she asked them to say them in different emotions, right? And so these actresses are probably saying what? They're actresses and she paid them, and so they did okay, we'll do what you want, right? So they would say okay happy and okay sad and so on and so forth. And in fact, it turned out that minor thirds in particular were overrepresented in sad 2-syllable words. That was it, nothing else came out, but sad 2-syllable words have descending minor thirds, and that's how you make them sad, apparently. But that's a little weird, right? Because emotional music and speech have a lot of acoustic cues. We just mentioned tempo being one of them, but here's some stuff from a nice review from Joseph and Lauka from quite a while ago now, I guess, of the sorts of things that seem to characterize different emotions in both speech and in music. There's a lot of them that are similar, right? And if you look at the ones that characterize sadness, a lot of them seem kind of inconsistent with a bunch of minor thirds. Minor thirds is a pretty big pitch jump for speech. And if you're talking about, low fundamental frequency, little pitch variability, that doesn't seem like minor thirds. So that's kind of surprising. But it may be that many of those things can't really show up in a 2-syllable word like okay. And in fact, in some work that we did during the pandemic, when we couldn't collect real data, we got a hold of, this is my postdoc Andres Buchalugo, we got a hold of a big database of acted emotional speech This is Frank Russo's RAVDES database. And they're neutral things too. They're kind of silly things like kids are talking by the door, but they're a little longer. There's a little more opportunity to do different kinds of expression in them. And actors said these in a whole bunch of different emotions. And so we just went through and we characterized all the pitch intervals in those spoken acted sentences in that whole database. When you look at the sad speech, you find minor thirds are not like especially common, but they're not like not that common. And anyway, the claim isn't that minor thirds are like in every sad speech, right? Bernstein's claim is that they're going to be overrepresented, but they weren't. So here you find that actually the most minor thirds showed up in disgust and anger, some in happiness, sadness not particularly, right? So that's surprising. I mean, again, I think what this goes to is that sadness is characterized by sort of flat pitches, and so minor thirds aren't that things like disgust and anger involve a lot more pitch movement, so you're more likely to get minor thirds because they're relatively larger pitches. But it doesn't mean that they're not important, right? Minor thirds do seem to be overrepresented in some contexts. So certainly there's the one I just told you about, which is when you only have 2 syllables to work with, apparently you might use a minor third. Here's another one. I bet you money that if I took a class of these same undergraduates and tried to get them to produce minor thirds, they would not do it as well as they do it when chanting airball. But that's a good minor third, so that's sort of interesting. What's that doing? It might be a broader instance of what people sometimes call a calling contour. So these are going to be like situations like saying dinner or like nya nya, right? Like where you sort of have these this particular kind of stylistic way of speaking that seems very often to involve minor thirds. Marco Polo, you can sometimes get this if you're like trying to get like somebody's attention, like excuse me. It turns out these are also very common in knock-knock jokes. They're in fact so common in knock-knock jokes that I found this little figure is from, not from research on minor thirds or anything like that. It's from a tutorial on how to speak English like a native speaker for non-native English speakers. And it has a whole section on how to tell a knock-knock joke. And the key is you have to say knock-knock on a minor third, or everybody will know you are not an English speaker, right? Because English speakers say knock-knock on a minor third. That's the story. Okay, I mean, so that's interesting. The minor third is doing something here, but it doesn't seem to be about like sadness, or really maybe about emotion at all. What's going on? What I think that this means is that the minor third in speech is just this sort of a toy example on meaning, but isn't really like an inherent musical linguistic mapping. It's like flexible, creative, cross-domain borrowing, right? You want to say, okay, sadly, what are you going to do? Kind of sing it, right? You want to like kind of use musical tools when you want to in language. It doesn't mean that they're doing the same thing from the bottom up, right? So I'm going to say that in general, where I am left, I'm sorry, Leonard, is that I don't think that the connections between music and language tend to be these really deep inherent mappings. I thought that for many years, and there's still part of me that does think that, but you go where the data tell you. Instead, I think what we really are seeing is shared reliance on underlying perceptual and cognitive abilities, along with some kinds of opportunistic cross-domain borrowing. Now, I think that's fine, right? I think that the understanding these cross-domain comparisons are still going to help us understand music and language better. I also think that they fit better with a little bit of a broader definition of what music is and what language is, because it's very hard to find very clear criteria for either domain, right? You end up with this sort of heterogeneous set of like, well, it's some combination of these things, some combination of those things, except for in those cases. And I think that thinking about this in a little bit of a more reductionist way of like, what are the underlying processes you need? What are the demands that different kinds of musical systems put on processing or different kinds of linguistic systems put on processing will hopefully tell us more. Thank you. That's my lab, by the way. They're great.
Hi, thank you for this. So you mentioned this example of musicality, potentially impacting speech processing, and I was thinking of the reverse of, what about speech processing affecting musicality? And it makes me think of tone languages. I think there are some studies showing that, speakers of tone languages, might be better at pitch perception or other kind of musicality related aspects. So is it meant to be a two-way street, or is it one way? Is there any reason that musicality to speech processing should be stronger than the other way? What are your thoughts on that?
Yeah, no, that's a great question. You're right. I mean, there are some kinds of findings that suggest that tone language speakers differ in some ways than non-tone language speakers in terms of pitch. I think the most robust finding there is that tone language speakers have a tendency to have higher prevalence of absolute pitch, which might, under this sort of more basic underlying stuff framework, might just mean like, look, if you attend to pitch a lot, you might attend more to pitch, and that's going to like have downstream consequences. I think why people have tended to focus on music's impact on language, though, is because music experience, ability, training is more variable, right? Like, you can go out and you can find people on a pretty big spectrum of musical ability. It's harder to do that in language, right? You can find different kinds of languages, and I think there's interesting work there, but it's a little bit harder to, so it's just sort of a practical reason, I think, right? If you want to say, I want a group of musicians and non-musicians, it's a little harder to be like, I want a group of like good language and bad language people. You can, depending on what you want to do. But I think your basic insight is exactly right. You'd expect this to be a two-way street. Yeah. Okay, thanks.
Great. Thank you. So now we are moving to our next speaker, Takako Fujioka, who is an associate professor of music at Stanford University. Her research topic includes neural oscillations for auditory perception, auditory motor coupling, brain plasticity in development and aging, and recovery from stroke with music-supported therapy. She did her postdoctoral work at Rotman Research Institute, and her work was supported by the Canadian Institute of Health Research. Her research continues to explore the biological nature of human musical ability by examining brain activities with non-invasive human measures such as MEG and EEG. Well, let's welcome Takako.
Okay, hi everyone. It's great to be here. It's my first time visiting LA, and it's a beautiful campus. And thank you, Asal, for inviting me as part of this amazing symposium. So I had a really hard time to compose this talk first, and then what are the common themes that I've been doing with various students and various topics, and what is this? And then I was agonizing over these things, and then I have to send the abstract to Asal, and then, please, and then I had an epiphany. This is about the multiple copies of the same information. And then, what does it mean? I will get through it, but it's really different kind of topics I'm dealing with because my students have various interests. But then this is all about something like how our mind, our brain deal with this abundance of confusing information and giving different kind of cues or mixing. Mixed messages, and then how the brain makes sense out of it. So here's the title. So the first talk I want to present about is the sense of beat. So Jessica did a great job in talking about the sense of beat and the rhythm perception and how the brain works. And recently I tried to think about where's the beat if complex music happens? So, you already heard lots of musical rhythm perception and the beat perception. And then there are some researchers talking about the poor synchronizers. The people can entrain to the simple metronome rhythm, but when it gets real music, or a little bit complex music, then it's really confusing for them, and then you can't really tap the foot or bounce to the music. And then also one of the actually Jessica studies individual differences is interesting. So even the people hear the same type of rhythm, exactly identical rhythm, but the interpretation of that rhythm is a little different. So that there's an individual variability to this. And well, come to think about it, we heard a wonderful music performance today. Are they playing really together? Yes, they are, musically. But if you go into look at the recording, the onset is having a synchrony of 0.000 millisecond, every single note. No, they're not, right? So what is happening? Music contains expressive timing. It's always variable. And they never play isochronously, right? And the musicians in ensemble, they're not strictly synchronizing. So what are they? What are we doing? So the first study is asking how much temporal spread we can tolerate, and then how do we integrate? So this study is done with one of the doctoral students at Kama, and to illustrate that point, I will show you this 100 metronome piece. So I'll go like this. Long introduction. Okay, do you hear beats? Sometimes a little bit, sometimes not. It's very confusing, isn't it? So it's in your mind that makes sense out of this pretty confusing situation. So we try to use a coupled oscillator network. First proposed by Kuramoto. It's a connected oscillator, limit cycle oscillators, and then we could use this bunch of oscillators connected with the various strengths, coupling strengths, and then make it tightly coupled or loosely coupled. So in here, okay, let's use this thing. In here, you are seeing this very strong coupling. So it's tight, and the sound onset here is pretty similar to the single metronome. The next one is a little bit spread, so it's loosely coupled, right? Or you can still see the silence in between, but it will sound like a brr, brr, brr kind of thing. Okay, so the sound waveform might look like this. Okay, next, more and more spread. And then in the end, no coupling is introduced. Now oscillator is like similar to this metronome. It's went ahead, it goes around, and then it keeps doing its set to do. So it's random spread, and then you will see the bunch of short intervals over here. Now we use that sound with various tempo and instruct people to tap wherever the beat you can feel. We didn't even specify you have to tap isochronously or whatever. They'll be free to do anything they feel like to do, okay? This is the chart we map in x-axis stimulus phase coherence from weak to strong coupling of the stimuli. And then here is how the tapping interval fares compared to that. So it's normalized the base tempo. So when we do the clustering, we see largely 3 clusters. So one is here. The coupling is really strong. So everybody's tapping to the bass tempo, no problem. You saw that before in the slide, right? Now the blue ones, even the coupling is weaker. People stay in there around the tapping interval of normalized 1. But you can see this one, this cluster, people produce much shorter tapping interval. So we call it frequency or fast tapping. So we try to go back, who's doing which tapping mode, okay? So we looked at each individual of those different strengths of the coupling, and then determined if one person doing more than half of the time stick with this regular tapping, we call it a regular group, okay? And then, then if someone do the opposite, more than half time, they do the fast tapping, we call it fast group. And the rest is hybrid. So now I'm showing the phase coherence circular plot in 3 groups. And then strong, medium, weak, none is on the column, 3 groups over here. We find the regular group to be like 34% or 17 people out of 50. And then when you see the strong coupling condition, you can see the tap timing concentrate around the tap, the stimulus concentration. So it's really following the stimulus density modulation. You see that? So the blue dots are people's tapping, and then red dots are stimulus. So blue and red nicely concentrate around the angle, right? Yes? Okay, I see notes. All right, so you can see the regular group now on the top. So they stay on. You can see slight concentration over here. Very nice. And then none, they finally succumb to the random distribution. First group, on the other hand, they lose the beat already at the medium. Okay, there's no density modulation anymore. They're already random. Okay? So when you see the 3 groups vertically, then you can see the tapping coherence decrease. Okay, so that's really interesting way to look at the individual difference. Now we're gonna look at how they start to find the beat. So I'm showing here the standard deviation of 3 consecutive tap interval. It's very difficult to maneuver this one. Sorry about that. Yeah. Okay, go. All right, so consecutive 3 tap interval. Now you can see regular group and a hybrid group. The least variable and really nicely settling it down from the beginning of the sequence, the strongest coupling condition. And then, of course, weaker conditions, the adaptation speed is slower. But you can see that none of them actually happens in a fast group. Okay? So, not only the phase coherence of the tap timing, they are very different in overall temporal maneuvering of making sense out of beats, or 40 metronomes, so to say. So our guess is that time course of adaptation and phase coherence led let us assume that there's some biological temporal constraints for the integration of auditory object. So that's interesting. Also, interestingly, the fast group, we find 9 people out of 50, and that 18% coincides with the proportion of poor synchronizers found in 3 studies that when look at the 100 or more people, general tapping performance. So it's kind of conspicuously same proportion. So we're currently following up on this studies, and then hopefully I have more neural data to explain some of that stuff. Okay, so quest continues. Question, how does the brain integrate multiple versions of information? We're going to switch to ears. Sense of sound in space. So if we have exact copy of the same sound in the left and right ear, of course you know that sound location cue is missing. So it doesn't happen in the natural world, but it happens a lot when you're hearing some kind of virtually configured sound. So your perception is that sound locates almost as if inside your head, okay? If we introduce a little delay in one ear, that would create a sense of false but very solid experience. The sound is being outside of your head. We call it externalization, okay? So, but you don't have any solid sense of where that is. It's just the outside of your head, okay? So any kind of, you have like a synthesizer or keyboard or whatever, something, you hit like a reverb, and that's what it does, right? Now, what happened when the sound is actually in the space? So what happens is that in the near ear, amplitude is larger, of course. And then in the far ear, not only the reduced amplitude, the sound arrives, but arrival time is delayed, right? Because sound wave has to travel a lot more distance to the far ear. And then you have a solid sense of where that sound is. Of course, that's not all. Physical shape of ear, head, shoulder, neck, all kinds of stuff will filter your sound in a different timbre. So you will have some sense of sound direction. Nevertheless, the acoustic cues that tells about the sound location is the tiny delay, especially in the lower frequency, interaural difference, and the amplitude asymmetry, especially in a high frequency, that gives you interaural level difference. Well, we speak, play, and listen to music or hear any kind of sound in a various space. So not only sound location, we're hearing everything with this information. It could be karaoke bar, it could be virtual reality, it could be in your shower room, right? And then in our institute, we have amazing 55 speaker surrounding system that lets a lot of creators and composers to play around and then include the spatial information into musical expression. Now, what about the musical experience? Some pioneering work show that the musicians have advantage in sound localization. So in this study, people have to say which speaker the sound came from, okay? And the only conductors, not pianists, not musicians, only conductors have the lower error rate. to be able to say, it is this or it is that, not in this speaker location. Well, it makes sense, right? Conductors are always looking at all these musicians, 80 people in the stage, and then direct them what's happening. So yeah, that's pretty obvious. Recently, psychoacoustic study congregating really whopping more than 200 people's just noticeable difference in ILD and ITD, and compare who has the advantage. Actually, they were able to show only extreme musicians have the lower threshold of ILD. But extreme, what extreme? Early starting and actively playing. So if you're a quitter, or you start late, then you don't have this advantage. Interesting, right? So where are we going? The one thing we didn't talk is reverb. That we have the space, we're hearing all kinds of sound, and then sound comes to you through direct sound path. But indoor space has all kinds of another set of reflective paths. Okay? So you have the direct sound arriving, first arrival, and then all the reflection arrives later. Okay? We have numerous copies of delayed sound information. It's diffused, it might be spectrally different, but anyhow, we're living in the sound space like this. Okay? So our question is, what's happening in here? The psychoacoustic studies showed the precedence effect, it's called. So what it says is that basically, listeners rely on the first arriving sound location through direct paths. But sound goes everywhere and give you all this reflective input. So do we get confused? Actually, listeners don't get confused. Listeners have the sense of one object despite the multiple copies, and it's called the fusion. And coming from the different locations, these conflicting location cues are largely ignored. They retain what they first find out, and then they ignore everything else. However, what happens is the echo's too loud, with too much delay, they fail to integrate this object into one. So they start hearing multiple sound. Does it make sense? Yeah? Okay? So most of studies use tone and clicks, and we're interested in how to, sorry, what is this? Sorry, how to externalize the given sound we have. So we collaborated with Elliot Canfield-Duffield, who was then a Karma doctoral student, and then introduced the group delay in real music, 2-second made speech, and country rock band song. We had 5 different frequency band and introduced the delay. So what happens in the delay, how much you can hear it? Actually, it's frequency specific. So as the higher frequency goes, and the GNT goes down. Oh, that's good way to find a sweet spot. And then what happens to the audio quality? So you can increase actually by introducing delay. You can introduce this spaciousness, but spaciousness increases as the delay increases, but then the quality of the perceived sound quality goes down. So it still gives us a really good information about the up mixing, and then make it artificial reverb, and then use it for compositional or mixing techniques. Now we want to know what happens in the brain. So I worked with another doctoral student, Maurice Vanzo, and tried to understand the brain processes where the sound location and the reverb information converge or diverge. So we use the MMN P3A complex to investigate this effect. And in reverb cues are small or big, different music practice space, and then position is front center or the right side front. So we could use the tone sequences, and then in this case, small serves as standard, big reverb serves as deviant. But at the same time, we can introduce position deviant, so front center all through, and then occasionally it's going to go to right. But the trick here is we can introduce both deviant at the same time. So we ask a question, is reverb processed independently from sound location, or they share neural resources? Sorry, it's kind of typo. But it is kind of, I was happy to see the Venn diagram that Bob was showing, because I'm going to show Venn diagram. So if two processes are independent, you will see the neural response to the both deviant that would be identical to the summation of position and the reverb. If they are sharing the resources, you might see the smaller response for the both deviant. Or maybe some other boost happening magically that gives rise to the synergetic, some kind of boost about the two information together. So we did that, and then we have the high tones and the low tones, and we tested the ERP. Here's the results. So I'm showing the front central 3 scalp areas. Now we can see that the pink line, which is summation, and the green lines is the both deviant response. In the MMN latency, you can see it's almost identical response in the both deviant and the summation. But in a P3A, both deviant gives you larger response. So the P3A, it's attention orienting and conscious registration of that deviance that's strongly affected by both cues changing together. What about the high tone and low tone? You can see that the low tone elicits far better response, and that tells us ITD cues may be more saliently connected to this neural resource sharing and enhancement. All right, so that was interesting, but that was only listening. What happened to the musicians? So we're going to go into, look at the brain process that is related to the reverb and other action planning, and then how that state of brain processes reverb information. So we're going to enlist the help from Schroeder. Schroeder is a pianist, of course. So he's always playing Beethoven, right? Okay. He's going to form an action plan. What not to play next, right? In his brain, the action plan is formed in a premotor cortex, but the premotor cortex is sending the predicted auditory image to the auditory cortex. It's called the efferent copy, all right? Now, he's going to execute the action. The piano makes the sound. He will have the direct path. He will have the sound feedback. But hearing the feedback, it's going to be compared against the efferent copy. So, if I tweak the feedback, something will happen. Brain doesn't like it. prediction error happens and then leading to the brain response, right? Well, that's good, but what about the reflective sound? The shredder might be in his room or he's playing a piano in the stage. So we're going to ask a question. Is reverb processed as part of action plan? Okay, so we have two master students at Kama and Kama executive director, right? And we're going to look at the hypothesis. Musicians may monitor auditory feedback holistically, so the two acoustic features like pitch and reverb might be processed differently alone but integrated together, okay? So including us, we are able to see the feedback-related negativity and the P3A complex when we throw in a wrong note. So the pitch is different from what you play, and then you will see this wonderful ERP. So what happens when we change the reverb? Okay, so here's a task. We have the two isochronous quarter note tunes, and the pianist has to play this over and over and over, and time to time we have pitch deviant, reverb deviant, and both deviant, okay? So, again, Venn diagram. Independent, sub-additive, or super-additive, okay? We also did a behavioral analysis around the deviant note, what happens. So usually if you hear something you didn't expect, you will have some kind of disruption. So we clearly instructed pianists, okay, no matter what, please keep going, right? But it doesn't mean that they are successfully ignoring everything. So what happens to their timing? So now, if big is serving as a standard, and then a small reverb happens over here, the deviant note becomes shorter, okay? And then next note go back up as a compensation, kind of cool. Now, what happens in a small reverb serving as a standard, it's opposite that's happening. So the deviant becomes longer, and then next two note goes down, okay, as a compensation. As you can see, it's interesting. Pitch deviant didn't really elicit any kind of tangible difference like this. They get a little upset, but not much. And then as you can see, the note-to-note intervals are longer to begin with when you have longer reverb. Okay, so from this, we can say that, hey, you know, small reverb makes pianists rush into the next note, and what big reverb makes them to wait. And then at the deviant, it happens exactly that too. So we can say that the reverb information seems to be encoded as part of the action plan for note duration. What happened to the ERP? You can see that the both deviant and pitch deviant is almost identical ERP happening. And the blue line is the reverb deviant is a little delayed. Nevertheless, if we look at the summation comparison, we have sub-additive this time. So it seems like pitch content matters more for the pianist. It makes sense. We don't want to hit the sour note during the performance, but brain does integrate reverb information in a performance monitoring process. Okay? All right, so last installment of the Sound in Space series is the auditory visual integration. So if a person is presented visually, oh yeah, you're gonna hear the sound in church. can you imagine the sound played in church, cathedral? Maybe you can, because you have enough experience and a memory about it, but is that memory only related to the bigness of the sound space? Or does it involve the type of places and the architectural complexity to it? We know that from the literature, people adapt their speaking or playing optimal for that space, and the room size translate to long and short reverbs, and then people have quite a good memory about it. So when we present exact match sound, people will be happy. But if I show the picture of a practice room and you hear big reverb from cathedral, it will be like, whoa, what is that? That's really bad mismatch. But what about this one? You're hearing the cathedral sound, long reverb, but you're seeing the concert hall picture. What happens? Do they differentiate these things? That's a question. So we have the concert hall impulse response, and then we can convert the dry sound. It doesn't work, does it? Okay. This is dry sound. This is impulse response. This is a playback, okay? Now we're gonna have the series of pictures and then sound combinations in a rating task. We have 6 big and 6 small spaces, half and half familiar and unfamiliar. We tested the Stanford student, and familiar spaces are all the Stanford music concert hall or practice rooms. They are pretty familiar with that. And then the other unfamiliar stuff, we rely on the other impulse response library. Here's the results. People rated highest of the exact match. And then between category mismatch, it's the lowest. But the remarkable thing was that the within category mismatch, concert hall and a cathedral, or dry recording studio and a practice room, they actually rated much lower than the exact match. So they actually have a good idea that's actually mismatch. Okay. Exact matching trials, the familiar spaces, big or small, they have more confidence in rating higher. The between category mismatch, it's interesting if visual big combined with audio small, the unfamiliar space has a little bit of inconfidence in the listener's rating. So from this rating study, we can say that the listeners have a really good idea of the indoor acoustic space, how that sounds like. Not only the size, not only the longest duration of the reverb, but the acoustic features of that kind of room. And then use that knowledge to imagine the sound from the picture, and then compare that against that memory. So that's kind of really interesting. We're writing this up. So here's a summary of the space perception. Multiple sources of information give rise to the sense of space. Delays and reverberations are perceived in a frequency-specific manner. Listener's brain processes spatial sound information together with locational reverberation. Musicians use acoustic space characteristics in auditory feedback for creating action plans. And listeners have internal representation of stereotypical acoustic spaces related to both auditory and visual information. Okay, so I'm at the very last topic here. I'm excited to talk about this new study. It's about the sense of pitch, but we're continuing the multi-sensory integration and perception. This time, audio tactile. Can people implicitly learn pitch tactile spatial pattern association? Would it help cochlear implant listeners to perceive pitch better? So actually, we heard all wonderful things about the cochlear implant listeners from Ray's presentation today, and then it's actually done with collaboration with Ray, Matt Fitzgerald at the Stanford Audiology, and a couple of Commerce students. So the previous studies that inspired us to do this is that the benefit of rhythmic cueing in making speech phoneme and a musical note onset, if you do that with tactile stimuli, then it helps people to perceive the speech or music when the audio signal is degraded. All right? So we use the 4-channel compressed air tactile stimulation. We have the plastic membrane and mounted to the fingertips and make the patterns, like discrete patterns, conveying the pitch class information of the major scale note. So we use the principle of general ascending type of placement, so do, re, mi, fa, sol, la, si, do, and then it's going like sort of digit 2 to from digit 2 to digit 4, digit 5. Sorry, I have typo here, but exact pattern of the finger mapping is not disclosed. They're not supposed to know this explicitly. We wanted to test whether that helps implicitly. So we did 2 studies. First, it's a proof of concept. Normal hearing listeners had a melody recognition task with the original audio, and then pre and post, we have the 2-tone pitch discrimination task. But that was not very difficult for them, except for that the audio is always degraded in a pitch test, okay? So I can play this melody. Everybody's probably know this melody. And then what happens is that the tactile stimulation will come with these patterns at each note onset. So and so on. All right. So this was successful. So we went on to test the cochlear implant users and the normal hearing listeners. In this case, we used the learning phase with degraded audio. So for the CI users, they use device. So we present the audio as is, but they get what CI gives them, right? But for the normal hearing, we have this cochlear implant stimulation. Well, you might say, well, rhythm gives away, but then we have another stimuli like this. Sorry. I want to go back. It was this. Or, oh, okay, so that's like that. I have a maybe have a label now. Okay. So maybe so rhythmically it's the same. We have the other, melodies that are rhythmically unique, but anyhow, we have enough foil stimuli. All right, so we tested CI users and then a normal hearing participant. So here's the results. I'm out of time, but it's almost there. Experiment 1, you can see the pre and post performance in a pitch discrimination task. You can see that the post test, everybody had improved performance in a pitch discrimination test in 3 modalities. Experiment 2, the great news is that CI users also improved not only audio tactile, but audio only. The normal hearing people did well too, this transfer. So they had a much better post-test performance at the larger interval size comparison. So we have immediate benefit and then transfer effect. We're trying to elucidate whether audio tactile performance is predicted by audio alone or tactile alone. So we conducted a regression analysis. As you can see at the pre-test, audio-only performance do not predict audio tactile performance at all. The model isn't significant. So that's why these are dashed line. But tactile-only performance, it's really one-on-one prediction to get the audio tactile stuff going. So that's great. Tactile augmentation is working immediately. That's what the literature says. But hey, post-test, what happens in the CI group is remarkable because only them change the slope. So what happens is that the audio tactile performance is now most steeply relying on the tactile information. They are forming the association and then using that information for audio alone condition. So that's great news. So this is the last slide, that's all. Thank you. We're chasing down what brain does or mind does in our perceptual process when dealing with the multiple copies of information. And we rely on these copies and then effectively construct some kind of perceptual image through integrative and sometimes inhibitory processes. Such experience can be implicitly learned and stored in the memory, activated on demand. However, inter-individual differences in perception may also arise from biological predisposition rather than experiences. That's it for today. Thank you so much.
I'm curious to know if you've noticed any gender differences. We know women have a satellite center in the brain. I think it's Wernicke's. And our spatial, visual, spatial, pupillary distance is different. So we perceive things differently. And emotionally and our cognition centers might be different. But did you see anything like in the spacing and other parts like that?
Yeah, in our studies now, but you brought up a really good point because spatial information processing is related to gender difference. But I believe that this kind of perceptual experiment, you need like probably close to 100 participants to look into that gender difference. Yeah, but thank you for your question. Yeah. Okay, thanks.
Wonderful. So for our final talk, we have a talk by Psyche Loui, who is an associate professor of creativity and creative practice from Northeastern University. Northeastern. bad. I was distracted by all the things you're interested in, which are listed here. The neuroscience of music cognition, musical perception, pitch problems, singing, tone deafness, music disorders, and emotional impact of music and voice. These are part of the interesting questions that Psyche explores in her research. What happens in the brain when we create music? What gives people chills? Can music be used to help with psychiatric and neurological disorders? And many more. Psyche graduated from the University of California, Berkeley with her PhD in psychology and attended Duke University as an undergraduate, graduating with degrees in psychology and music and a certificate in neuroscience. She has since held faculty positions in psychology, neuroscience, and integrative science at Wesleyan University and neurology at the Harvard Medical School, and she's currently at Northeastern. So with that, let's welcome Psyche.
Thank you for your kind introduction, Asal, and thank you for organizing this wonderful, wonderful day and inviting us. So it has turned a otherwise somewhat depressing week into a really wonderful gathering of friends, old and new. So I'm very excited to tell you a little bit about some of the newer projects that my lab's been involved in. But like a lot of the theme you've heard throughout today, music is something that is celebrated in all human societies. And it is something that we use to express passion, as well as intergenerational care. And I don't have to tell you to rock out to a concert for you to want to do it, right? So much of what we know and love about music seems to be something that's implicitly learned. And you might think that musical ability or musicality, the ability to perceive and produce and appreciate music, is something that's uniquely human, but it turns out that it's not even uniquely human, because there's this famous parrot called Snowball, the African gray cockatoo, that has been known to dance Backstreet Boys, Backstreet's Back All Right. And if you speed up the Backstreet Boys, it speeds up to dancing, and if you slow it down, then it slows down its dancing. So it's actually beat extracting, and it's not just imitating. But I wanted to start my talk with a different kind of music-induced movement, and it's the ability to be moved by music emotionally. How many of you have felt one of these kinds of intense aesthetic responses to music? And I see a show of hands. Raise your hand if you've ever gotten chills for music. How about if you've ever felt like music made you feel like you were somewhere else? Who has ever felt like crying when listening to music? Wow, that's just a really quite a very strongly hedonic group. So this is just a smorgasbord of a few different strong emotion or aesthetic responses to art that people report, and predominantly people report them mostly for music. But we also know that, so most people, when confronted with intensely emotional situations, like Demi Moore in Ghost, who saw the ghost of her dead boyfriend, might show some strong emotional responses, like tearing up. But then there are also some characters who, despite being very cognitively intact and intelligent, just don't get emotional for anything. And you might think that that tracks with gender, perhaps, but it turns out that that doesn't always, right? So a few years ago, Matt Sachs, who's here in the audience, did his undergraduate thesis with me, where he started to ask why, right? Why are there some people who get chills all the time when they listen to music, and other people who don't get chills at all when they listen to music? And so he painstakingly recruited students, Harvard undergrads at the time, who were, we'll call them the chill group, who often report getting all of these very strong, intense sensations. And then another group that was matching for musical training, matching for gender, even matching for personality differences, who just don't get chills when they listen to music. And we used a special type of magnetic resonance imaging, or MRI, that Peter introduced earlier, called diffusion tensor imaging, that's especially useful for looking at white matter. And in this case, we're interested in white matter connections between the superior temporal gyrus, which is a part of the auditory network, network in the brain and the anterior insula and medial prefrontal cortex that are together parts of the reward and social networks. To make a very long story very short, what we saw was that the chill group, right, the people who tend to get strong, intense emotional experiences to music, had larger larger patterns of white matter connecting these auditory and reward networks in the brain. So this was our first foray into brain structural differences that underlie this aesthetic experience. But when that paper got published, I was at Wesleyan University at the time, this gentleman got in touch with me. We'll call him BW. And BW has what he has found to be musical anhedonia. So he enjoys good food, he enjoys social input, he enjoys a back rub like any of us, but he just doesn't get it for music. And it's not that he is deaf, right? It's not that he has any music perceptual difficulties, but we interviewed him at length and he would say things like, well, I would stand in the checkout line for a drugstore and I might see some teenagers who are bopping to music in their headphones and I would wonder, why would you want to do that? Which to me seems both sad and also very intriguing. So we tested him at length, and musical anhedonia is something that is just becoming known about at the time. And the gold standard neuropsychological test at the time was called the Barcelona Music Reward Questionnaire, where you're given a variety of questions asking for things like music seeking, right? How often do you look for new music, and also how often do you seek out music that your friends also seek out? And so it characterizes individual differences in the sensitivity of rewards of music listening along 5 different dimensions. And BW turned out to score more than 5 standard deviations below the mean in all of these 5 dimensions. We also tested him in a different neuropsychological test called the physical anhedonia scale. So we asked things like, do you enjoy the sounds of organ or the sounds of leaves rustling in the trees? Or do you enjoy good food? Or do you enjoy long walks on the beach? And it turns out that BW was no different from the control group in all things except for sound-related items. Right? So while he was just as likely to enjoy a long walk on the beach as you and me, he just did not enjoy even sounds of leaves rustling in the trees or the sound of ocean lapping on the shore, which are things that other people generally report finding pleasurable. And again, to make a long story short, we looked at diffusion tensor imaging of BW's brain compared to a large group of age-matched controls, and we saw that BW had different pathways of white matter connecting the superior temporal gyrus in the auditory network and the nucleus accumbens, which is part of the dopaminergic reward system. So based on the BW's work and a few other papers that were coming out at the time, our colleague Amy Belfi and I sat down and looked at and tried to list all the brain regions that are together part of our neuroanatomical model for music and reward, right? So areas like the superior temporal gyrus that are part of the auditory network might need to be connected to areas like the medial prefrontal cortex or the dorsal and ventral striatum that are together part of the reward system, and those 2 networks need to be intactly connected for sensitivity to musical reward. And maybe if you have a lesion or a different altered neural connectivity in any of these regions, then you might end up with musical anhedonia. So that's just on paper. And then we had another undergraduate student, Diana Wang, who took these regions of interest and actually translated them into a brain atlas. And so now we have a set of regions of interest that we can use as hypotheses for functional magnetic resonance imaging studies like this one. Right? So in our lab, we've developed a music listening functional MRI task where people bring in some pieces of music that they select for themselves. And then we also have other pieces of music that we select as researchers, some of which are well known to them or within the culture, and others that are unfamiliar. And I'll talk a bit about both more in a bit. And in all cases, people listen to these melodies one at a time, and then they had to rate how much they liked each piece of music, and they also had to rate how familiar each piece of music was. And we did this in young adults and also older adults. And what we saw was that, here I'm just showing you the main effects of music listening in the scanner during music that you subsequently rate as hated or neutral or liked or loved. And you can see that in general, can you see my, in general, the superior temporal gyrus in areas that are part of the auditory system are generally active, even for hated music. But there's additional activity in the medial prefrontal cortex when listening to music that you love. And this is true both for young adults and for older adults. However, if you look at the functional connectivity, right, so this is now trying to take these regions of interest that Diana had identified and identify their pairwise connectivity, as in the correlation of activity between different pairs of regions within the auditory network, within the reward network, and across the auditory and reward networks. And when we did that, we saw that within the reward network, young adults showed higher functional connectivity, so the reward network was better intrinsically connected for young adults than for older adults. And then furthermore, for auditory to reward out-of-network connections, especially when listening to music that you love, young adults showed this advantage of stronger functional connectivity between the auditory and the reward networks, right? Suggesting that this selective functional connectivity between the auditory system and the reward system is something that changes with age. Right, so functional MRI, we've already heard a lot about functional MRI today. Thank you, Jess, for that super wonderful one minute summary of what that is. But we also know that the other thing we've heard a lot today is EEG work. Right? We also know that functional MRI is relatively temporally sluggish, whereas EEG is more temporally accurate. And so what we also wanted to do was characterize the way our brains oscillate in time to music as we're listening to that music, right? So again, in young adults and older adults, we have people bring in music that they choose to listen to for themselves that they like, and so we know that they're going to be engaged by it, and then we simply record EEG while they're listening to this music. And our analysis is a little bit different from the event-related potential types of studies that Takako and Peter showed, but we specifically looked at phase locking, right? So taking the amplitude envelope of the music and correlating that with the EEG data in a phase-by-phase cyclic sort of way. And importantly for this paper, we normalized across different pulses, right? So we know that natural music unfolds in many different tempos, some of which is faster and some of which is slower. We did not instruct participants to bring in music of any particular tempo. We just took whatever the tempo was, and then we matched their beats per minute. Right? So, and so this pulse normalization method was able to, we were able to use that to show phase locking value in a way that's centered on the pulse, regardless of what frequency or what the tempo of the music is. And when we did that, we saw that both young adults and older adults showed this hierarchical phase locking between the EEG and the acoustic signal. That centers around the pulse level, right? So the optimal finger tapping tempo of the music, but we also saw additional phase locking in the harmonic and the subharmonic of the tempo. And furthermore, we thought at first that older adults might bring in less beat heavy or beat salient music, or that they might show different patterns of neural entrainment, but both of those didn't turn out to be the case. It turned out that older adults were just as likely to bring in music that had strong rhythm in it, and that they were just as likely to show this entrainment as young adults. So this was pretty exciting to us, but now we wanted to see if the difference in auditory reward functional connectivity that I've just presented from the functional MRI data might be something that you could intervene on, right? So we worked with neurologists, Maya Geddes, who's a geriatric neurologist who specializes in the reward system, who's now based out of McGill University, was at Harvard at the time, and then also Suzanne Hanser, who started the music therapy department at Berklee College of Music. So really a card-carrying music therapist. And we all worked together in this multidisciplinary team to design a music-based intervention where older adults came into our lab and worked with a music therapist one-on-one, where they curated, co-created music listening playlists that were then listened to every day for an hour. And for each day during that listening, the older adults also wrote about their reflections in a logbook. So it was a mindful music-based intervention that required thinking about the music and also being receptive to it. And every week, the therapist would call these older adults and check in with them and see how they're doing and if they wanted any of their music changed. And of course, before and after, we did a number of intervention, pre and post-intervention assessments that included different kinds of MRI and also neuropsychological testing. So there's reams and reams of data from this study that we're still analyzing, but the original hypothesis was that there might be some change in auditory reward connectivity when you look at seeding the auditory regions of interest that I've shown you that Diana had identified. And so what we did here was we took the auditory network and we asked our MRI data, where do you see any changes in functional connectivity from the auditory network to anywhere in the brain? And when we did that, the only region that popped up as being statistically significant after controlling for multiple comparisons was in the medial prefrontal cortex, which is, of course, an integral part of the reward network. And furthermore, that sensitivity of the medial prefrontal cortex was especially stronger for self-selected music relative to other selected music. So this shows us that there's something about the auditory engagement of the reward system that changes throughout the lifespan, but is enhanced by this mindful music listening intervention. So we thought this was pretty exciting, and this is a kind of baseline for a music-based intervention that we're going to keep working on and keep designing. But I wanted to start to answer one of the kind of higher level questions that was raised earlier about why we have music in the first place, right? So, and I was part of this multidisciplinary collaboration that kind of put out this position paper a couple of years ago. That's to say that musicality is something that co-evolved as a part of gene-culture coevolution for social bonding. So basically, we have music because it brings people together. Specifically, there's something about musical features like beat and like meter, but also like harmony and melody and scales that together tap into the prediction and reward system, and that is how music brings different people together. So I think there's a lot there, and a lot of different ways to sort of tease apart the questions that this model kind of brings up. But I just wanted to focus a little bit on the bottom part of that, and especially I wanted to look at how these cultural constructs like harmony and discrete pitches might tap into the prediction and reward systems of the brain. So just a little bit more about what we mean by these discrete pitches and scales. So what do we mean by musical scales, right? So musical scale is defined as a range of pitches that are discrete, but that have predictable features, right? So musical systems around the world are composed in scales that usually range in a 2-to-1 frequency ratio, right? So the octave is based on a 2-to-1 frequency ratio. So one tone that is one octave above another tone that is 220 hertz or cycles per second is twice that, which is 440 hertz. And then within that 2-to-1 frequency ratio, the equal tempered Western chromatic scale divides that ratio into 12 logarithmically even steps, right? So this is what a equal tempered chromatic scale sounds like. Okay, and you can compose different pieces of music by using different steps along that scale. So for example, the Western diatonic scale can be used to give rise to something that sounds like this. So that was written by a French composer trying to sound Spanish. So other cultures use other steps along that scale, but all cultures use the 2-to-1 frequency ratio, right? So for example, some of you might know this piece. Same violinist playing the same instrument, but it's a very Chinese pentatonic scale. So even if you haven't heard that piece before, you might notice that there's something about the use of that pentatonic scale that signals a kind of very Chinese identity. So neither of these cultures used a scale that's based on a different integer ratio altogether. So Heinz Bolin and John Pierce were two engineers that independently started to ask, why are musical systems based on 2-to-1? Why can't they be based on 3-to-1? Right, so they proposed this alternate scale, and that's based on a 3-to-1 frequency ratio, which we'll call a tritif instead of an octave. And then within that 3-to-1 frequency ratio, it's 13 logarithmically even divisions. So it sounds like this. And here is a piece of music written in that scale. What do you think? Does anyone imagine some stories when listening to that? Does that sound weird? Does that sound good? Does that sound oddly otherworldly? Oddly otherworldly. Okay. Right. So the reason why 13, right? Some might ask, and it turns out that if you take that 3-to-1 frequency ratio and you divide it into 13 logarithmically even steps, some of those pitches circled here approximate low integer frequency ratios. So they sound more like stable chords. And so you might be able to hear, even from that piece, reminiscences that I just played, there are some tone clouds that kind of emerge that are designed to be relatively consonant together, even though they're different from consonant chords in the Western sense. All right, so I thought this was really fascinating, and what we started to do was take these different chords and string them together in a little bit like a finite state grammar, and so we can now have a chord progression. So this is a 4-chord chord progression in this new scale. Okay, and so now we have a chord progression, and we can start to compose melodies using this new scale. So Yiwen Zhang was an undergraduate in my lab, and he wrote this melody. And because he wrote this melody, he can also change it so that it's slightly different towards the end. And you could tell that the two melodies are quite similar until the very end, and the contours are the same, and most of the intervals are the same, but it's just that that second melody doesn't, quote, go back to the one chord, right? It doesn't return to how it started. And so because we can generate these melodies from scratch, we can also have a lab-based experiment where people would come in and listen to different numbers of melodies, different numbers of times, and so we can systematically manipulate both the frequency, as in number of times you hear each melody, and the transitional probability, as in the likelihood that you hear one chord given another chord just before it. And so these can be independently manipulated. And so what we did was just have people come into the lab and listen to a bunch of these melodies different numbers of times, and then afterwards rate each melody for how familiar it sounded and how much they liked it. And so just to show you these are across I think 600 different participants based in the U.S. And we saw that people were very much likely to rate melodies that had been presented more as being more familiar, but the melodies that ended differently were rated as less familiar. And then more interestingly, we saw that reward, right, liking rating, how much they liked each melody, also scaled with the number of presentations and then decreased with the prediction error, right, with the different endings. We also did this with a couple of hundred people based out of China, right, with the idea that they might have different levels of exposure to different scales. And what we saw was that it didn't matter. So the Chinese folks behaved much like the US folks in that ratings for both familiarity and liking increased with number of presentations and decreased with the prediction error. But one group that didn't behave similarly to the US and the American folks were the BWs of the world. So people who scored low on the Barcelona Music Reward Questionnaire, actually, they still reported being familiar to the melodies as much as anybody else, but it took them much fewer repetitions before they said they didn't like it anymore, right? So suggesting that there's something about the mapping between prediction and reward that's disrupted in people with musical anhedonia. Now, finally, recall how I told you that when I was introducing the functional MRI task, that some of those melodies were unfamiliar. So we snuck some melodies in there that were part of the Bohlen-Pierce scale. And what we saw was that when listening to melodies that are in the original system, the superior temporal gyrus and Heschel's gyrus was more active in response to the original melodies relative to the altered melodies, right? So suggesting that it's coding for prediction. And then the functional connectivity between the superior temporal gyrus and Heschel's gyrus and the medial prefrontal cortex also increased with the number of presentations and then decreased with the prediction error, mirroring the human behavioral results, right? Suggesting that this mapping between the auditory and the reward system is what gives rise to how novel predictions become rewarding, or how music acquires its reward value. So that was really exciting to us. And around that same time, we also learned about some work by Lisa Margulis' lab at Princeton, where they had taken pieces of music that were either from a Chinese culture or from a Western culture, and asked people to imagine stories for them. That's why when I played you that reminiscences piece the first time, I said, do you imagine anything? All right. And so what they saw, this is a very kind of snapshot of their results, is that these people that are Dong people, they're not even Chinese people in China, they're like a specific group of people that are in this very remote village called Demon. And those folks, turns out they still imagine stories when they listen to both Chinese and Western music. However, the semantic distance between Chinese listeners for Chinese pieces was closer to each other, whereas the Western listeners reported stories that were more semantically close to other Westerners. Right? So even though both groups were listening to the same set of melodies, they formed different imaginings that were filtered through their culture. So I thought this was very cool, and I said, Lisa, let's have some people listen to Bohlen-Pierce melodies, right? And so what we did was we have a few hundred Bostonians and Beijing people listen to melodies that were composed in the Bohlen-Pierce scale, and then afterwards just imagined stories and just wrote down stories that they imagined for each piece of music. And here is a smorgasbord of the same piece of a word cloud of the words that were reported in the stories that were reported by, that were generated by that Stephen Yee reminiscences piece that I just played you. So in a nutshell, it seemed like the Bostonians reported imagining someone walking in the dark in the ocean, whereas the Beijing folks reported imagining a man in the night slowly walking through a forest. All right. But then what we did was we can quantify the semantic similarity for each piece of music within the culture and between the culture, and still the Bostonians imagined stories that were more like each other, whereas the Beijing folks imagined stories that were more like each other, right? So there was a larger semantic distance between cultures compared to within cultures, again, replicating the results that Lisa's group had found before. And another interesting part of this result was that we could also, given that each person can have can report a story that can be characterized as a certain point in semantic space, we can actually treat this as an individual difference, right? So let's say if you're listening to one piece of music and you're generating a story, right? And then the center of all people's within one culture, all the stories that are generated by that culture would be the center of that semantic space. And it turns, and each person's story can be characterized as one point in this space, such that people who are maybe more unusual might report a story that's a little bit further away from the consensus, whereas people who are more prototypical, right? Let's just say they're more influenced by the popularity contest, right? They might immediately report the story that's close to what everyone else was saying. So we wanted to look into this as a measure of individual differences in creativity and imagination. And so this is just quite kind of hard to see, but this is showing that for each song, for each piece of music, the number corresponds to the semantic location of the consensus narrative across people, and then each dot is one person's report, right? So there is quite a lot of spread, even though the narratives are also quite clustered. And it turns out that this distance, right, the distance of, from the semantic mean was correlated with in absorption subscale of the Barcelona Music Reward Questionnaire. So people who reported being more emotionally engaged by the music tend to report stories that are closer to the consensus, whereas people who report having a good ear, right, so perceptual ability, which is something that you could measure using a scale called the Goldsmith's Music Sophistication Index. So people who had this higher perceptual ability by self-report actually showed an inverse correlation, inverse relationship with the semantic consensus. So people who are sort of, shall we say, highly trained conservatory type musicians might actually be so busy thinking about whether that note was out of tune that they don't actually imagine the same stories that everyone else seems to be imagining. All right, but so that was an interesting foray into music-based imagination. So given all that I've reported about Bohl-Pierce scale, I also wanted to see whether people can actually create new melodies from that scale. So we designed a task that's run using MaxMSP. We asked people to generate different melodies in the Bohl-Pierce scale. And this is a very useful tool because you can start to quantify how predictions map onto reward in a way that's different from more standard studies in musical improvisation that usually rely heavily on training. Right? So now we have a relatively training-independent measure of musical creativity. And so what we did was we asked people to generate as many melodies as they wanted using this interface that were in the Bohl-Pierce scale. And then we asked another group of participants to listen to those melodies and rate them for how creative they were. So we have this generation and evaluation loop, akin to this perception action loop that Peter presented earlier. And so in general, and the other nice feature of using Bohl-Pierce music as a source of as a test for musical creativity is that you could very conveniently apply musical informatics as predictors for creativity ratings. And what we saw was that melodies that are longer are rated as more creative. Melodies that have more unique pitches in them are rated as more creative, but there's a linear and a quadratic relationship there. And then melodies that have higher interval entropy are rated as more creative. So with more complex intervals comes higher ratings of creativity. So here I'll just play you a few of these example melodies that were generated by our participants who were otherwise not musicians. So that's a fairly typical melody that was generated and was rated as kind of medium levels of creativity. Here's a very highly rated melody. And then finally, the next melody I'm going to play is across a database of 700 or so melodies that our participants generated. This is one of the lowest. You can kind of tell that it has low entropy and low length. And then importantly, what we saw also was that people who were able to generate more melodies, longer melodies, were also more likely to do well in a classic creativity task. And a way to investigate creativity in a non-musically dependent way is something called an alternate uses task, where you list all the uses you can think of for regular household objects like a brick. And people who made longer melodies also came up with more uses for a brick. Right? So suggesting that there's something domain general about this way in which we're assessing creativity. All right, so I've talked a lot about using weird scales to measure creativity and to measure learning and to measure reward. But I also wanted to leave you with some more applied work. Right? So what is this good for? And maybe by now you've been hearing me blab on for so long that your resources for sustained attention is quite waning. Maybe you're starting to mind wander a little bit and check Facebook or Instagram or whatever you have. Maybe you're starting to think about the clouds and generally phase out. And that's okay. It turns out we all do that for about a third of our day. But maybe if you actually really want to focus on this, or maybe you actually find yourself mind wandering in an unhealthily large amount of your time while you're actually supposed to be being productive and studying and reading and doing your work. So is there a way in which music can help reduce our mind wandering and focus our sustained attention? So a couple of years ago, we started to hear from this company called Brain.fm that claims to be an auditory neuroscience startup. And what they claim to do is they claim that they compose music that entrains your brain waves. Right? So we've heard quite a bit about entrainment today already. They claim that they're making music that helps you focus. And I was at first quite skeptical, but there's plenty of rave reviews on social media. And I do think that there are, from what we know about neuroscience, there is something about these neural oscillations, or the ability of different regions to synchronize together in time, that subserve different modes of brain activity, and that together subserve things like sustained attention and mind wandering. Right? So there's this really beautiful review by Kalina Christoph et al. That talks about how goal-directed thought is a set of constraints in your mind and when you have some self-generated thought that have certain automatic constraints that are not under your own control, you might start to do things like ruminate and have obsessive thoughts. Whereas, on the other hand, you might have more deliberate constraints that are under your own control, might be useful for things like mind wandering, but are also creative thinking. Right? So the same set of neural oscillations, depending on how you tune them, might be important in some cases for goal-directed thought, and in other cases for creativity. So BrainFM's CEO was like, wow, I want to use this to make Netflix for the brain. And I was like, well, come on, let's just test this first. All right, so what exactly is their music doing? So let me just show you some visualizations. So BrainFM thinks of Spotify as one of their competitors. We know some Spotify workers here. So this is a piece of music that was downloaded from playlists that Spotify put out for study. study music. And so a lot of undergrads use this for working in a library or whatever. And so I'm going to show you the music that is going to unfold here in this spectrogram-like manner. And then at the top, I'm going to show you what's called a modulation spectrogram of the music. Right? So the amplitude modulation, the rates of that amplitude modulation are represented on this axis. And then in the middle, all you're seeing is a summation across the different cochlear channels in this amplitude modulation rate space. Okay, so this is what Spotify's music sounds like. All right, so you can see how as soon as the music starts playing, this starts scrolling, and then the top also starts changing around, but they are not really clustering in any noticeable way. So compare this to Brain FM's music, right? So I'll just play it first. Yeah, so in a nice reverberant environment like this, it's a little bit hard to tell what's going on, but the visualization does make it clear that the amplitude modulation is unfolding at a very relatively fast rate, so in the beta band actually, so the 12 to 16 hertz range. And you can see how there's acoustic energy that's centered around here. Right, so if you just take a snapshot of the amplitude modulation spectrogram, you can see how Brain FM has this very focused amplitude modulation in the beta band around 14 hertz range that's not observed in the other ambient study music. So what we wanted to do was we wanted to test, we had people listen to these different kinds of music while they were doing a sustained attention response task, which is a fairly boring task that induces mind wandering that I'll show you in a moment. And here I'm showing you the phase locking value, right, that I'd already talked about in the earlier slides. And you can see how while the brain is generally phase locking in lower frequencies to in the control condition, the amplitude modulation added music shows some very specific peaks of phase locking to specific frequencies. And so we also have people listen to this while doing this fairly boring task called the sustained attention to response task, where numbers would show up on the screen one at a time, and you just have to press the space bar for every single number, except for if it's a 3, in which case you don't press the... because this is quite boring, after a while you might start to mind wander, and then you might start to make a false alarm, and that results in the oh shit response that Peter introduced earlier. And we did this in people who, and we also asked people to self-rate the numbers of symptoms of ADHD using what's called an ADHD self-rating scale. So just had to rate how often they experience certain symptoms, like how often do you have trouble wrapping up final details of a project once the challenging parts have been done? Or how often do you have problems remembering appointments or obligations? Right, so people with more subclinical ADHD symptoms are more likely to self-report on the higher parts of that scale. And it turns out that that's quite a lot of people. Almost about 40% of our participants end up scoring quite high on the ASRS scale. And what we saw was that this stimulus brain coupling actually was generally cleaner at first in the low ASRS group than in the high ASRS group, right? So the people with more ADHD symptoms show noisier phase locking to the music. However, if you break down the data over time, comparing the early part of the recording and the late part of the recording, the higher ASRS group actually shows an increase in phase locking over time, suggesting that the music was better able to tune their EEG for it to work. And so I was pretty excited about this. Kevin, the first author of this paper, was even more excited about this. And so what he was able to do was systematically insert these amplitude modulations on otherwise the same piece of music. And we had people listen to these different rates and depths of amplitude modulation and do the sustained attention to response task. And what we saw was that in general, the modulation rate was helpful for higher ASRS folks, right? So people who have more of these ADHD symptoms tend to do better over time when they're listening to the beta band amplitude modulation in the 16 hertz range. And then furthermore, they needed higher amplitude modulation depth, right? So we could, the more deeply you apply the amplitude modulation, the more the folks that had these ADHD symptoms would be helped. Yeah, so I think that's pretty exciting. We're still following up and doing more work on this idea of using new music for amplitude modulation. And I'll just take my last minute to wrap up. I think there's something about looking at new music and new musical systems and new musical experiences that can help us understand how sounds tap into the reward system, right? So I've shown you the special case of musical anhedonia, which is an abnormal auditory prediction to reward mapping. And these predictions unfold over both spectral and temporal domains, and they originate from statistical exposure to sounds. And what we also saw was that there's something about rapidly learned predictions that can be applicable towards understanding musical creativity and imagination. We have a few projects that are more applied work in music cognition that takes these new musical experiences and tries to tailor them for special populations like people with ADHD. And I didn't get time to talk about it, but we're also doing some work on Alzheimer's disease. And generally, I think there's something about musically induced rhythmic entrainment and phase amplitude coupling that might be viable mechanisms for predictions that are sustainably able to engage the reward system. And that might be how one way by which music-based interventions might be useful for cognitive and brain health. I think I'll stop there and thank the wonderful students and the lab members that made this work possible, and also our funding sources that made this possible. And I'll also finish by saying this day has been really wonderful to see different people from different disciplines coming together, and we have a book that just came out last year that really is a set of essays that are a dialogue across humanistic and scientific approaches to music. It's freely available on open access on the MIT Press website. Feel free to take a picture of that QR code, and both Asal and Matt, I think, have book chapters, and also Beatrice as well. So yeah, I think I'll stop there. Thank you very much.
Time for one question.
Thank you for the great talk. In terms of the amplitude modulation being at around the beta band, is there kind of a specific reason for that, or does that work better than other frequencies? And do you think it could be related to like beta oscillations in the brain, which are prevalent in motor areas?
Yeah, I think it is very much related to beta oscillations in the brain that are prevalent in motor areas, and like Takako has done some work on that. So when I listen to that music, right, the Brain FM music, I feel like I'm really energized, even though I'm not using my hands, right? And so maybe that's what is going on. Maybe it's activating the SMA, right? So some of the same networks that we've been hearing about all day. But I don't know that it's specific to beta. So we're actually now testing theta band amplitude modulation, as well as inserting gamma band energy using visual stimulation and multi-sensory stimulation as well. Thank you. Thank you.
And with that, thanks to all the speakers for traveling and for sharing their work and expertise with us. I think it was such a great day. We learned so much. And thanks to HCN for co-sponsoring this event. And thank you all for being here all day. I hope you had a good day and wish you a wonderful weekend. Thank you.